I've started the recording. Well, hello and welcome everybody to the fourth instance of the machine learning for drug discovery event. This time around, we're going to have a very nice session, I believe, with great amount of very, very exciting speakers. So super happy to have you all tuning in today. I just want to give a quick intro to the event itself and the schedule before we dive into the first session. And for this, I wanted to briefly walk you through what the machine learning for drug discovery event is. The Machine Learning for Drug Discovery, short MLDD event, is an annual event happening every year ever since the 2021 instance, and is now actually in its fourth year already. And really, we see it as a gathering of industry professional, academics, and enthusiasts for drug discovery and AI. And we want to really ensure and use the mission of it to ensure an interconnectedness in the field and surface the best and most interesting ideas that may ultimately help develop medical innovations that have a high chance to make it to patients in the future. And we're super excited actually that we have a number of very, very excellent speakers that we were able to win over for this event this year. We'll be joined later today throughout the day by Charlotte Bunne from EPFL, John Chodera from Akira and MSKCC, Kexin Huang from Stanford University, Jacob Kimmel from New Limit, and Professor Marinka Zitnik from the Harvard University. All we believe covering a nice cut through the field of AI for drug discovery and some of the most exciting ideas and directions that are being worked on today, from virtual sense to AI scientists to neural network potentials to knowledge-driven and knowledge-informed drug discovery. I think we've covered it all in this instance, so I think we will have a nice and exciting afternoon here with these sessions. I just quickly want to walk you through the schedule of this event today. All the times in this schedule are in the UK local time, and we'll be starting today with a quick introduction, which is this session, just introducing you to the event itself and the schedule. And then later on, we will be diving into the actual keynotes, where we will be starting with Charlotte from EPFL, going later towards Marinka, joining us from Harvard University, John from Akira and MSKCC. We're also planning for a short break, understanding that it might be quite knowledge and information dense to walk through these sessions today. So we'll have a break around 3 p.m. UK for around 15 minutes until we rejoin with our West Coast speakers, Jacob Kimmel joining us from New Limit, and later Kexin Huang from Stanford University will also be part of the event. The last part of the event will actually be some closing remarks and also the survey results. And the survey is actually a new element of the MLDD that I also wanted to quickly introduce to you. And also, in case you haven't voted yet, it's certainly a good time to vote now, perhaps. The inaugural MLDD survey will actually try to answer some of the most interesting and exciting questions of the field from your perspective. So we're really inviting all of you to give us your opinion on some of these questions, including, are we close to bio-AGI? What is the most promising research direction in machine learning for drug discovery? And what is the greatest bottleneck for impact and achieving impact with machine learning for drug discovery? There's lots of controversial questions as well, which I think we will have some fun time discussing the results of towards the end of today's event. This is the first time that we're doing this, so please bear with us if there's any like operational challenges around this, but we have posted the link to the survey on the ml4dd.com website. And in case you have not voted yet, we've seen that already a lot of you have voted, but in case you have not voted yet, your last chance is throughout the session. So please feel free to have your opinion reflected there and vote in terms of what you believe might be some of these answers to some of these exciting questions so that we can discuss them and we have a good reflection of the entire community in that survey. Towards the end, we will have a little bit of time in that last half hour to look at the results, investigate what that might mean, and just hypothesize about these results. So I think it will be exciting if all of you contribute to that and show your opinions in that survey as well. Lastly, before we get started with some of these initial sessions, I also wanted to let you know that beyond the stream that we're doing right now on YouTube, where we're broadcasting the event itself, we also plan to make available the recording of this event after the fact. So we will be posting that recording on the ML4DD YouTube channel as well, so that you can watch it afterwards. So no worries if you cannot make all the sessions today, or if there's some conflicts that you're facing today. Eventually, we will make available all of the recordings as we have in the past instances as well, and we will post it to the ML4DD channel on YouTube and advertise it as well for you to revisit those talks that you may not have been able to join for the session today. And so I hope that makes it relatively convenient for you to access the information that will be discussed later today. Lastly, I also wanted to acknowledge the many contributed and many people behind the scenes that are trying to help us make this workshop and event happen, showing some of the faces here that have been kindly supporting us with the organization of this event. That will be Stefan Bauer from Helmholtz and TU Munich, Florian, formerly of Novo Nordisk, Arash joining us from DSK, Marcel from Oxford, Pascal currently at Harvard, and myself and Martino as well from DSK, Mathieu, ETH, and also DSK, and Francesco joining us from Oxford. And I just wanted to acknowledge and recognize their kind contributions in making this event happen behind the scenes. Obviously, a lot of work to get such an event off the ground. Now, I think we have around 5 more minutes until the event gets started. So I would say we wait 2 more minutes and then we start to introduce Charlotte here, and we will start just around 1 p.m. mark. So we will be rejoining and restarting and revisiting you just in a few minutes. So bear with us as we get set up with the first speaker, and see you then.
Hello, Charlotte.
Hey. Do you hear me? I think now I hear myself as an echo.
Yeah, can you hear me?
I can hear you, yeah.
Perfect. So welcome, Charlotte. It's great to have you today. So I'm going to introduce our first speaker, Charlotte Bunne. She's an assistant professor at EPFL in Computer Science and Life Sciences Department. Her research focuses on applying machine learning to personalized medicine and drug discovery with methods from optimal transport, control theory, dynamical systems, and diffusion models. Before joining EPFL, Charlotte was a postdoc at Genentech and Stanford. She completed her PhD in computer science at ETH Zurich and also did research at the Broad Institute of MIT and Harvard. She's a recipient of the ETH Medal and a fellow of the German National Academic Foundation. Charlotte, it's great to have you today, and please take the stage.
Perfect. Thanks so much. I assume you all see my screen?
Yes, we can see.
Perfect. Great. Thanks a lot for having me. I'm very happy to speak. So I'm going to talk about virtual cells and digital twins, AI in personalized oncology. So this might be a very familiar site. It's been a staple of modern oncology, or something that's been almost around by a century, that gives us an idea about a tumor's architecture and overall state of or pathology of a tumor of a patient. And what we have seen over the last decade is that this world of spatial biology has become much more colorful. We are now able to really look at molecular properties of individual cells and often in an iterative fashion basically highlight many different proteins at almost a subcellular resolution. So here you can see many different cycles that all highlight basically many different proteins and their intensity across a tissue. And so this spatial biology is one staple, besides many others, that allow us to really look into a patient's profile or molecular profile to be able to understand the state of the disease, possible diagnostics/prognostics, what to do, how to proceed with the treatment, and so on and so forth. So on the one hand, of course, we have a classical histopathology, but we also have what we call spatial proteomics as well as spatial transcriptomics. Besides, of course, all the genomic profiling, besides records, lab tests that we can collect treatment history from a patient. And we are growing larger and larger atlases of such data or cancer types that really make us go one step further and ask how can we use AI-based predictions that are operating across these very diverse and heterogeneous modalities to get the best representation and understanding of a patient's biology, and then ultimately this treatment and treatment strategy. And we are working very closely with molecular tumor boards. So this is a board of doctors consisting of oncologists, geneticists, pathologists, and many other experts that need to basically go through a decision process for every individual patient. And here incorporate a lot of different modalities. So we are operating heavily multimodal as well as longitudinal over an entire patient history that can sometimes span almost 15 years or longer, right? So when a patient actually has a recurrence and the cancer comes back. Besides, of course, all the information that needs to be incorporated that we know from the literature, all the knowledge databases that are available, bioinformatics tools, ongoing clinical trials, and so on and so forth. The questions we are asking is how can we basically support this decision process using AI-powered tools that we are developing in the lab? And those questions might be very diverse. It might range from cancer staging, therapy, response prediction to predicting a clinical outcome, and so on and so forth. But also more that allow us to get a better understanding of the underlying biology, different states, different cell types that are present, and the interaction in a tumor microenvironment that might lead for resistance to a treatment or particular sensitivity. So on the one hand, we have a lot of different tasks. So we are sitting in a multitask, or we want to build models that are task agnostic, which we can use for many things at the same time. And we are operating on very different modalities. So we are on the one hand, multitask, multimodal, and the AI methods that we develop should facilitate exactly this transition. The question is how, right? In histopathology, we actually have seen fairly successful histopathology foundation model that can do some variety of tasks. And those models usually basically are built on really large compendia of databases of histopathology images. So here we have really images spanning many different cancer indications, as well as kind of hospitals, cancer types, and so on and so forth. And they're usually trained in an unsupervised fashion, where we crop the image into many different tiles, and we get some internal representations that we can then use to basically predict here pan-cancer detection, subtyping, biomarker prediction, and so on and so forth. But the moment we move to the next modality here, spatial proteomics, we are already getting into a situation where we cannot predict very reliably because we don't even know how to deal with the data in the first place. Histopathology, these are RGB images, so a data modality in computer vision that we are very, very familiar with. Spatial proteomics, on the other hand, is a multiplex image. Every channel here is the presence of indicates the presence and the identity of a protein, often subcellular. So these are the colorful images that you saw at the very beginning of the talk. And we have many channels, sometimes up to 150, that can come out of one spatial proteomics experiment. How do we build architectures, right, or foundation models with this input? It's not clear. We don't have an architecture, so we really need to think about new architecture developments that allow us to do basically this multimodal understanding of the patient's biology. We need to have methods, AI tools for all these different data types. And if we look at what has been really driving the success of AI in, for example, natural language processing as well as computer vision, it's really architectural principles such as like attention, self-attention, CNN, RNNs, as well as their basically kind of aggregation into entire architectures such as here vision transformers or kind of NLP transformers that has been really driving the development. Besides those architectures, of course, the like kind of overall objectives that we are using to really train them unsupervised or in a self-supervised fashion. In language, right, this principle of predicting the next token, like the next syllable or the next word, is something that really allows us to understand the overall structure. The context window is quite small, right? Our sentences are quite short. That works very well. Same for masking in computer vision. It's a very good principle to teach an algorithm the overall structure of images and how they are met. Like if I mask a part of a dog, it's relatively straightforward if I have enough training examples to fill it in. We need to find those building blocks, architectures, and objectives also for biological data. And this is exactly what we were thinking of when we were writing this piece on AI vertical cells. Like, how do we build basically AI architectures that allow us to really encompass the complexity of biology that operates on the molecular scale into a cellular scale and multicellular scale. So basically where we have data that we are collecting here on the molecular scale, RNA, DNA, protein, and we can build really architectures for this, but they need to be connected, right? We need to tokenize those entities and connect them to the next level where we have then basically information on the cellular phenotypes and then of course tissue phenotypes. There's also this challenge of being able to really predict dynamic processes in biology. And so this AI virtual cell also has some proposal how we can build AI architectures that really capture underlying dynamics and dynamic processes on every different scale that we can parameterize. And that's exactly the goal of this talk, to give you some idea on how we build such architectures, right? How can we think of new design principles that allow us here to basically predict tissue biology, or like kind of like to predict tissue, like kind of extract information from tissue. And the second part of the talk, we are talking briefly about generative AI methods that allow us to really to forecast dynamic changes here for tumor cells to predict the response to a therapy or drug. So let's get started. One of the staples of the Swiss Personalized Oncology Initiative that I'm closely working with is multiplex imaging, here in particular IMC. So this is it gives us in the end a multiplex image where every layer, every channel is indicating the presence or absence of a protein. So we have intensity values across here a crop out of a patient's tissue. Every channel is a different marker, and depending on the clinical study or the problem at hand, we might record a different set of channels. It's not like in computer vision where I always have the same RGB. It's not always RGB, same input, same channel, same combination of channels. No, here I might have 40 to 100 channels. Here you can see this for basically 5 different data sets. Every data set has a different combination of channel and a different number of channels. The data sets that we work with are pretty small because it's always clinical studies which have like varying number of participants, sometimes 10, sometimes 1,000, right? But we need to combine them all to really train those foundation models on very, very diverse data sets. We have a lack of clinical labels, so we need to operate here unsupervised. And right, we have a heterogeneity in the study design, so we need to come up with an architecture that allows us to operate with a varying number of input channels across many different data sets. What we want to build is some form of virtual tissue representation that we can now use. Basically, I can embed a patient's biopsy into a virtual tissue representation that I can now use to predict clinical diagnostics, as well as information about like biological discovery tasks like cell type annotation, niche annotation, and so on and so forth. Ideally, we would like to construct this virtual tissue database. I mean, here we do this Swiss-wide, but ideally we do this internationally, where I can now look at a patient who had a very similar molecular profile. And I can query this and I can see what has this patient been treated with, what has been successful or what has been not successful. So I can really guide clinical decision support in a most useful and impactful way. What we want to build is a foundation model, meaning something that's trained and that allows us to do zero-shot inference. So for a completely new data set, I can compute a virtual tissue representation that are then used for prediction or information retrieval tasks. And we need to do this future-proof, right? Maybe in a future study, we are even operating on a completely new channel, a completely new protein. But as I said with this AI virtual cell, if we build our models multi-scale across the scales of biology, we incorporate a lot of information on the protein in the first place, we are able to go and operate basically on completely unseen channel, on completely new markers. So what we do is we need to find a new tokenization scheme that works for multiplex images. So we have basically here 40 different channels, for example, for a data set, where we need to incorporate the information of the identity of a protein, here 1 pixel. Here we use a protein language model that has been trained on the entire basically protein data bank and incorporates basically or embeds the gives us information about a protein in the first place. That's of course one way of doing it. There would be many better ways of also incorporating pathway information, literature information, and so on and so forth. But that's a starting point at least. And now I need to decode the basically identity of this protein at a particular spatial location. And this fusion of spatial and marker token is the now kind of new representation of my multiplex image. And this way I can incorporate whatever combination of proteins I'm measuring in a particular experiment, even if I'm thinking about constructing those minimum diagnostic tests, which we will just discuss in a second. And we need to build our model multi-scale as well, because we want to have readouts on the cellular level, on the niche level to understand tissue properties, and on the tissue level to understand the overall biology of a patient, or like overall decisions across on a patient level. We need to think really up to the building blocks, because now I'm operating on images that are sometimes actually 100 dimensional, like I have 100 different channels. As standard attention doesn't operate, it doesn't scale anymore to these large images. They are on the one hand really large and have a lot of different, like a lot of dimensions. So what we did is basically come up with a new form of attention, which is across markers as well as across space, which also has a positive feature of giving us some interpretability to highlight what are the most important protein markers, right, or potential biomarkers for a patient. What are the spatial properties that result in a certain decision? Here you can see the immediate effect of choosing this new attention. This is a standard vision transformer developed by Meta, which has been used to basically train on multiplex images. And you can see it doesn't scale at all, right? We stop at 40 channels because this is the size of some of the data set, but also it just gets very expensive to compute on a GPU. You can see how well our method scales because we really design new architectural principles for these data sets. At the same time, right, the more information I add, the more multiplex channels I have, the more information I get about my biological sample. And if I tell the model, like this is protein A, this is protein B, through our tokenization system, it really also is able to incorporate more and more information being added across the number of channels, like the more channels I add. That's completely natural, what we want to expect. Of course, it might saturate because this is enough channel information. We don't want to see like that more information actually hurts the model performance. That's exactly here, this meta vision transformer that's just not been designed for biological data. Here you can see visualizing on the one hand, the spatial attention, right? Here predicting cancer type, the model looks at the presence of the tumor cells here in dark blue and highlighting what are important markers for a particular tissue region. It's very interesting because here for the tumor markers, for the tumor regions, it's tumor markers. For the immune cell regions, it's immune cell markers. That's very, very nice to see. And then the moment we have patients for which we know they have responded and they have not responded to an immunotherapy, it allows us to really also look at the marker level, what's been the most important marker to distinguish non-responders from responders, for example. Why do we want to be zero shot, right? We might just have a new data set which incorporates a new marker and we want to still be able to embed it, right? And we want to be able to have a new data set and then still predict very, very well the cell types. You can see here, even if we work zero shot, we have a very robust prediction of cell types. The same holds for here tissue level where we want to predict relapse or mutation status, cancer stage, and so on and so forth. We want to be able to incorporate new markers, right? And this kind of you can see here, this is the embedding of the protein language model. And then we have in yellow, basically the new data set with a couple of new markers. But mLANA, for example, as a new marker is embedded very, very well. We've seen a lot of similar proteins, at least in terms of their sequence identity. And you can see that the moment we give the model some pixels, the reconstruction is basically perfect. But the interesting part is here, not giving a single input, really in painting or reconstructing this marker from scratch. This works well when we have it incorporated into the training set. If we don't have it incorporated into the training set, you can see that the overall structure is captured, but it's not able to get the intensity level right. And our performance degrades the more channels we add. We want to use this as a clinical decision support tool. So we want to be able to really retrieve previous patients that have a very similar tissue phenotype. Here, this is the input query. These are the 3 hits, all 3 adenocarcinomas, late stage, mostly no relapse. And this is exactly what we get, right? Same here, another adenocarcinoma apparently, but very different tissue architecture. You can see the queries we get, the candidates we get for a query, they really very much resemble the overall tissue architecture and also the meta information that we have. Here, no relapse, the first to have also relapse. It's again a grade 2 adenocarcinoma and so on and so forth. But we can start now, the moment we have those foundation models for histopathology, for spatial proteomics, really think one step further. The same way as we use reinforcement learning for human feedback in LLMs, we can start thinking about how can we reason or optimize whatever we measure and capture about our data. And here in particular, given such powerful foundation models and many different technologies, how can we optimally select and sequence diagnostic tests? Meaning, can I use them to, on the one hand, tell me which diagnostic test is needed in the first place? Histopathology is very much a low-cost, easy test. Spatial proteomics is, of course, very, very costly. Often these are good enough, but can the model tell me when to do an additional experiment for a patient? And if so, can it tell me the optimal 6 markers, right? Because now I have a model that operates on any combination of markers. Can you give me the optimal combination of markers that gives me the most robust virtual tissue representation that I can then use to predict different downstream tasks, whatever we are interested in? And we do this exactly in the same fashion as it's done for LLMs. We have a reinforcement learning system that basically sits on top of those foundation models for histopathology or spatial proteomics, and now allows us to basically optimize and for these different type of questions. So MAVIS is a reinforcement learning system that is multimodal and allows to find a policy that either decides when to measure some different modality, or within a modality, which type of features to measure. given some form of reward that optimizes for the cost as well as the accuracy we get, and the amount of uncertainty will reduce. So here, in fact, we ask an expert to give us the markers that they would select for spatial proteomics for a particular cancer type to predict. Here, this is, I think, cancer type prediction. And you can see that the first 3 markers here, this is selected views. Views is just a formalism to allow for both feature as well as modality selection. These are all different views on this, on the kind of problem. But then after the first 3, it's not any more clear, you know, what are the markers that really help to get a robust enough representation to get the highest accuracy possible. This is the, while at the same time, you know, reducing the uncertainty about this prediction. And we need to also do different modality selection, right? You can see just running histopathology on those type of cores, it gives us a prediction accuracy of around 75%. And here we get something like 85 overall when we run always a spatial proteomics experiment. But can I use, you know, only a spatial proteomics experiment, or a kind of suggest a spatial proteomics experiment only in cases where H&E would prove wrong, right? This is exactly what Marvis is able to do, right? It gets the same accuracy as always running a spatial proteomics experiment, but here you can see, and this is log scale, on average, the cost for a patient would be $50 because for most of the cases, we only require histopathology and we don't need a full 30-plex, like 30 channels measured, but often 6 channels, 5 channels is enough to get very robust predictions in the first place. So this gives you a bit of an idea on how we can build those foundation models across the scale of biology, incorporating information that is measured. Basically, if we measure here an image that measures a protein, we can give information about the protein, get those representations, and then also modules that optimize on it. As a very last part of the talk, I would like to briefly introduce these generative AI methods that allow us to also use those kind of representational states to then forecast how cells would, for example, respond to perturbation responses or any other kind of genetic chemical perturbation, whatever we are interested in. So ideally here we would be able to in silico model cell behavior, so models that really allow us to understand on the level of individual cells how a cell responds, for example, transitions from a healthy into a disease state or transitions the moment it's been perturbed by a drug. And what's important here that we want to do this on the level of single cells, because we know cellular populations are incredibly heterogeneous, in particular in cancer. And we want to be able to identify which part of the population is responding to the treatment and changes in which fashion, while another one is actually a resistant subclone that would require either a combination therapy or just a different form of treatment, or might then recur or grow after treatment. So we have a very quick resistance of this patient to the treatment applied. The challenge here is that every time we measure a cell, we destroy it. So we don't get single cell trajectories. We don't measure the same cell before and after. So we somewhat need to really operate on the level of distributions, right? To predict how an unperturbed distribution changes into a perturbed distribution. Now we have those embeddings, right? Either in a cell data space or in one of those embeddings, representation learning algorithms we introduced just before. We now want to find a map that really predicts how this maps a cell into its unperturbed state. So T of X would be then the unperturbed state of a cell X. We need to do this on the level of distribution, but we can find some hypothesis space that describes an optimal map T. And I think it's important to note here that usually those drug screens are run on a relatively short timeline, so 8 to 24 hours. So we can make the assumption that a map that overall minimizes the misalignment, so I find for each cell the most likely fit, might be a good prior and hypothesis to put in to really predict perturbation responses here. So what I'm looking for is a map that really minimizes the misalignment cost over all alignments. And that's a famous principle known as optimal transport, going back to Gaspard Monge. And we've also not been the first people to do this in optimal transport, but what we've really pioneered is methods like neural networks that really behave as an optimal transport map. So now here we have developed a generative model, like a generative AI tool, that now takes in a collection of cells and forecasts, given a drug, how those cells would respond upon basically perturbation with the drug. So we predict the perturbed distribution, but we can get for every cell now a trajectory on how they changed. And there's a series of methods that are all kind of connected under this principle of optimal transport. What I'm going to show you here in the next is not really a detail on how those methods work, but really like showcase them on how they're currently used in an observational clinical cohort. So this is methods that been published almost 2-3 years ago, right? But these clinical validations just take much, much longer. But by now, these methods have been almost superseded by tools that are mostly based on the fusion model of Schrödinger bridges or flow matching, however you call it. And so we have a couple of works that are really along this connection from optimal transport maps to the really nice theoretical connections of PDEs and dynamical systems, as well as control theory. So here we have those diffusion models, flow matching, Schrödinger bridges. And there's a couple of very powerful tools developed by us, but also collaborators and colleagues in the field. And we've summarized basically their connections in a tutorial. So this is with Marco Couturi, my PhD advisor at ICML 2023, it's still public, and where we really describe also the mathematical connection. And with Aviv Regev and my two PhD advisors, we've written a Nature Reviews Methods primer on all optimal transport and single cell and spatial omics. Now what we have is a cohort of patients, and we want to be able to predict for a new incoming patient, given their control cells, how they would now predict, basically respond to a drug. And this predicted treatment outcome, as well as the treatment effects, because we know how every cell changed, will now allow us to assess the success or the efficacy of a treatment in the first place. And we can see, because we have here an ongoing kind of observational clinical trial where we have actually 37 patients, and it's much more, but we used a subset of 37 here that were available at this time, for which we have data for a drug screen of 34 different clinically approved drugs. So now on the one hand, we can predict the treatment outcome for individual cells. Actually, I was not planning on showing these slides, but let's briefly walk through it. So this is for 2 different drugs, panobinostat and crizotinib, for 2 different patients, how basically for 2 different features. So we have a number of patients and a number of treatments times number of features, these type of plots. Here I just show you 2, because we want to basically forecast how those distributions change. And so here there's only a reduction overall in SOX9. Here you can see really a subset of the cells of this patient respond. So there's really a shift from one subpopulation to the left. And that's exactly what our method captures. While the other methods, especially used to be autoencoder that has been used for this, really just predict an average effect and they move everything to the left. At the same time, right, we also have an extension of this method that we now use, which is able to condition on everything in addition, because there might also be, of course, alternative factors and confounding factors that decide on decide the response of a patient, but we can condition on them, for example, driver mutation and basically previous number of treatments. And you can see here for dabrafenib, there's, for example, a resistance for some people who have a driver mutation. So those don't respond, while another sub-cohort actually responds. And this is exactly what a model that's not conditioned on this meta information would not capture. So you can see they predict no response here and a response here, but a method which is identical to CELLOR, just able to condition on this method information, is able to really capture nicely no response here and a response of patients who basically have this particular driver mutation. But what's important and what I really want to highlight is why we need to really develop methods for very specific tasks in biology, because now I don't have a method that just predicts the overall treatment effect or just an average effect. No, for every cell, I can subtract the perturbed from this unperturbed state and get how every feature changed overall. We know the progression of those patients, so basically how long they still survive with the treatment that they've gotten, and we know which treatment they've gotten ultimately from those 34 different drugs. And interestingly, to predict really acute or slow progression, it's not the accuracy on the meta-information, it's not the accuracy on the state of the disease before, on the state of the disease after, but it's really extracting how the patients, and in particular this tumor cells, responded to a drug that allows us to understand if this treatment would be successful, resulting in slow progression versus unsuccessful, resulting in acute progression. And this is a really like kind of innocent plot, but I think the biggest success moment of all those generative AI methods that we've developed that really allow us to forecast not only the effect of a drug, but also what it means ultimately for a patient's journey. And so here are just two ideas on how we can design those new AI tools for precision oncology. And these, of course, translate to many other domains in biology. And I think what I'm particularly curious now that we have more and more methods that allow us to be predictive as well as generative, to think one step further, how can we make those methods to be queried so we can extract the knowledge that they've incorporated and then in particular start reason and hypothesis test in an automated fashion that allow us to basically extract and discover new biological insights in the first place. And with this, big thank you to not only my PhD advisors, of course, still Andreas Krause and Marco Cotturi. So in particular, the second part was still part of my PhD. Also, my postdoc host, Aviv Regev at Genentech and Yuri Leskevich at Stanford, as well as my co-first authors, Yusuf and Yanai, with whom I've written the AIVC, as well as Emma, Steve, and he's not here, Theo. But then also my clinical collaborators at SGG, Olivier, and at University Hospital in Zurich, Andreas, as well as a very close collaborator of mine, Lucas. The first two works I presented were already part of my new lab. So we've been at EPFL now 9 months, and Johan and Ishan were the first authors of the virtual tissue, as well as the reinforcement learning package. So big thanks to them, and very excited to work with them for many, many years. So thanks so much, and happy to take questions.
Yeah, thank you very much, Charlotte, for a very insightful and comprehensive talk. Yeah, I have some questions myself, but let's start with the questions in the chat. So the first question is, what kind of approaches are you considering for mitigating the impact of biases like batch effects when predicting out of dataset predictions?
Yes. So, I mean, I think batch effects are just not to be avoided, but I think we can work on the data representation as well as also the objectives. So, for example, if I look at absolute values or relative values, right? So we can basically reformulate the problem in a sense that I don't look at this, I'm not regressing a particular, I don't know, RNA-seq count number, but I can just look at an overall like relative presence. This gene is much more often here than elsewhere. And this allows us to compare data sets. And then, of course, the more data we add, the more we should be able to also mitigate biases. And this is why we are developing methods that are so flexible in the amount of inputs they can take. So we can just incorporate much more data sets. So I showed you here in this talk 5 data sets. That's what we used to develop the architecture. Now we're actually training this on 20 data sets, and 20 data sets is pretty much every data sets that's publicly available, plus the ones that we have in the court. So I think just scaling as well as the architecture design allows you to mitigate for it. I think really the more data you add, the less you can do manual removal of batch effects. So I think just with the scale, this doesn't become so viable anymore. That's just So that's at least what we take. And in these multiplex images, batch effects are quite large, right? Like you have artifacts, you have really different intensity values. So so we do a really easy normalization and then just basically understand overall, how much of the result is biased by batch effects. And I mean, I think there's also we need to be realistic. In the histopathology foundation models, even though they've trained on a massive amount of data, you can still predict fairly well which hospital they were from. So I think we don't get rid of them yet. So I think also there's a lot of work that still needs to be done.
Yeah, great. The next question is related to the last part of your talk, optimal transport. So how do you evaluate the accuracy of the optimal transport maps when there's no ground truth at cellular level?
So we look either on distributional measures, right? Like, so how well does this distribution overall change? But this is indeed true that this might just be, if there's an oscillatory behavior, right? Those metrics might be perfect, even though we don't actually capture the fact that there has been some form of, like, I don't know, circulation, and we might be doing a completely wrong mapping. What we also did in one of the works was that we used data for which we have barcoded cells. So we knew at least that the cells, like the daughter cells of the cells, that we would map those kind of barcoded cells properly. So we had a data set for which we could do an evaluation based on this. But otherwise, it's not possible. I mean, I'm dreaming of a data set that gives us these like kind of like high resolution on a molecular level, and then live cell imaging, right, where you can actually trace cells overall. And if you have some form of good mapping of those, I think that would be a really, really nice starting point to evaluate also the trajectories overall, because right now we actually only hope for the endpoint to be correct, even though ideally we also want the trajectories to be correct.
Yeah. So one question that I myself had was, so when you define this optimal transport measure, distributional measure, you also need to define this ground metric, right?
Yes.
And the ground metrics is somehow a similarity, or your belief about the similarity of two cells, right? This is kind of a way to integrate your inductive bias about the similarity of two cells.
Quite tricky. So I mean, with the diffusion models, we started basically imposing, I mean, so there the default would always be just assume it's Brownian motion and the data space. One thing we did is that we've assumed, okay, so if I can at least find an Gaussian approximation, right, so that I move basically along an average with the standard deviation across, so I find at least I impose some path already, and then you can train this much more efficiently. That was one approach. I mean, there's also, you know, when we think now about the foundation model space, you know, maybe we can impose optimality in terms of the like kind of the manifold that's underneath. I think there's many things to consider. So far, we've always worked with very dumb priors because we don't have better ones. So for example, here with the single cell profiles, we are operating in an embedding space and using Euclidean distance, which just is not ideal at all. Yeah.
And maybe one last question from the chat. I think maybe that's also related to the earlier question. What is the process of fine-tuning of perturbation prediction models for unseen cell types? And what are the minimal conditions required for a viable model to do in silico drug screen?
Yes, so we are not imposing any information on the cell type, but basically the model would not, there's no guarantee that the model extrapolates to a completely unseen cell type. And this has been really done a couple of years ago. I think what I really also believe with this idea of an AI virtual cell is it's not that we are modeling one cell, right? Like it's more an infrastructure and how can we incorporate as much data points or measurements as possible to kind of constrain the hypothesis space. And if we have, for example, with the human cell atlas, you know, at least like a huge amount of different cell states covered, and we have a disease atlas. So, you know, maybe if we are building methods on top of those large atlases of data, it's our ability to generalize, like even if I have a new clinical cohort, right? But then our ability to generalize to those kind of unseen cell states should be much more robuster because they need to satisfy also the constraints of historic measurements. So that's a bit the overall idea. Our ability to do this should improve the more data modalities we add also, right? Like, especially if we have different data modalities that actually paired, because they both should give this, describe the same biological system, right? And should constrain the space our model fills in much better. Here in the results I showed you, that's I wouldn't dare to say or give me a completely new patient and we would be able to fit. Here it was even interesting to see which patients can be fit better than others, because these were actually patients with a, you could see very different like kind of profile, because they had driver mutation XYZ, or they had a very particular treatment history. And it was incorporated because this information was not in their single cell profiles, of course. That's why we even came up with the conditional model. So the conditional model was developed after we moved to the clinical cohort, because we realized we needed to account for this. And then there's this huge bulk of confounding factors that we don't incorporate, right? That, of course, still influence the treatment decision. It's really hard to really work out anything, any kind of influencing factor. So it's limited.
Yep.
Yeah.
Thank you very much, Charlotte, for sharing your ideas and your view with us. Very impressive work, and looking forward to more interesting works from your new lab at EPFL.
Thanks so much, and I guess we see each other soon in person again. Bye-bye.
Yeah, thank you.
Thanks so much.
Bye.
Bye-bye.
Okay, our next speaker is Marinka Zitnik. I think we can continue in around 5 minutes. So yeah, thanks, and stay with us. Hello, Marinka.
Hello, Arash. Can you hear me?
Yeah, perfect. So feel
Okay,
free to
great.
share your slide just to make sure it works.
Yes, I'd like to. I'm sharing my screen now. So can you see the slides?
Yes, that's perfect.
Okay, great. So I'll stop sharing now and then I can restart.
Yes, we will start in 3 minutes. Thank you.
Okay, sounds good.
So, hello again, everyone. Our next speaker is Marinka Zitnik, Associate Professor at Harvard Medical School and affiliated with the Kepler Institute, Broad Institute, and Harvard Data Science Initiative. Her research focuses on building AI methods for scientific discovery and personalized medicine. Before Harvard, she was a postdoc at Stanford and part of the John Zuckerberg BioHub. She earned her PhD in computer science from the University of Ljubljana and has received several honors, including NSF Carey Award and Kavli Fellowship. So in case you're following us on YouTube, please feel free to share your questions on the chat. So we will ask Marinka those questions at the end. And with that, please, Marinka, the stage is yours.
Hello, everyone. Thank you very much for this kind introduction. I'm delighted to be here with you today and share our research. Let me start by sharing my screen. Okay. So today I will talk about how we can empower biomedical discovery with AI scientists, and I will highlight several projects throughout this presentation that all come with open data sets, models, and evaluations linked from our lab website. Over the last several years, science as well as biomedical research has been transformed through tight integration of AI and machine learning models into scientific workflows and biomedical discovery. And generative AI is having an impact on science by both changing the way scientists go about their everyday research activities and also by providing insights that might not have been possible using traditional methods alone just a few years ago. In my group at Harvard, we are focusing on the paradigm of AI scientists, which is that of creating co-pilots that can augment and research done by human scientists and can collaborate with them, with the vision that eventually we will be able to build AI that's capable of skeptical learning and reasoning, that can easily integrate multimodal data with experimental platforms and learn and innovate on its own. And so to illustrate the AI scientist paradigm, in this talk today, I will highlight the research that goes towards realizing this vision across 4 scales of biology. We will start with models that operate at the level of molecules and atom-level representations of molecules, then proceed with modeling of molecular interactions. Those molecular interactions do not take place in isolation of each other, but instead they're embedded in molecular and cellular environments. And finally, we will look at the higher scale of modeling therapies and modeling treatment response in patients. So let's start with the first part, which is fundamentally driven by the question of understanding what do different molecules do and what their roles are in humans. So as a field, we now have models that can predict protein structure from sequence and do that so well that for many proteins, these models achieve atom-level accuracy. So this has been fantastic advance over the last several years that has to some extent realized the grand challenge of being able to better understand the structure for each and every human protein. Another grand challenge, however, remains largely unsolved, which is that of predicting protein function from sequence structure, as well as other protein data types. This still largely remains an open challenge. 20% of human proteins lack well-defined functions, even when well-annotated proteins often miss functional insights across biological contexts and disease states. Many human proteins are missing understanding of how their function varies with biological contexts, such as cell types and cell states. And a large fraction, over 95% of publications, focus on just a small number of well-studied human proteins, around 5,000 of them. So this creates a very big important challenge of how to be able to model, predict function for each and every human protein, and realizing that would be fundamentally important for various questions in drug discovery, starting with target identification, validation, and understanding. I want to share with you a model we recently developed of that kind, which is called Procyon. So Procyon is the first multi-model foundation model for protein function understanding, prediction, and generation. Procyon as a foundation model for protein functions allow human scientists to collaborate and work with this model through 3 different interaction interfaces that are shown on this slide. Human scientists can describe in plain natural language prompt the task they would want Procyon to solve, as well as provide in the input prompt description of biological function, biological process that they would like to retrieve proteins against and rank them based on how likely they have a particular function of interest. Alternatively, human scientists can interact with the model through question answering, where they can provide to the model in the input prompt protein amino acid sequence structure interleaved with text description of biological phenotype, and then the model will engage with the user in question answering. And finally, in addition to predictive modeling, Procyon is able of generating high-quality protein text or protein phenotype descriptions that can serve as captions of proteins based on interleaved multimodal inputs that are encoded in the protein in the user prompt. On this slide, I'm showing a high-level overview of the architecture of Procyon that we can read from left to right. So in the left, we can see how user can specify the variety of different types of queries in their prompt. And importantly, the input prompts are naturally multimodal, meaning that both in the input as well as in the output, users can interleave text with protein sequences and structures in any form and shape they would like. These multimodal prompts are then decomposed into 3 parts, where protein amino acid sequence is modeled by protein language models, the structure is modeled by atom-level protein structure encoders, and text is modeled by a large language model. These 3 data modalities are mapped into a unified space through the structure, sequence, and text projectors, and based on that unified latent space, the model either can retrieve proteins from its large protein library to perform search and retrieval against the user prompt, or the model can generate free-form text descriptions that are candidate phenotype descriptions or predicted protein functions that the query proteins likely have. To train Procyon model, we have created a new data set, which is called Procyon Instruct Tuning Dataset. Procyon Instruct Tuning Dataset consists of 33 million protein phenotype pairs that span 5 areas of biological knowledge, including protein functions, disease understanding, mechanism of action of drugs, protein functional and structural subdomains, and molecular interactions. In addition, there are over hundreds of millions of protein sequences, hundreds of thousands of structures captured in the sequence and structure encoders, and full-length papers for natural language understanding captured in the text-based LLM that underlies Procyon. To train Procyon model, we have implemented 3 key objectives. Specifically, we used the language modeling objectives for next token prediction. To elicit contextual protein retrieval behavior, we relied on contrastive learning objectives, and finally we used instruction tuning to ensure that the model follows human instruction provided in the input prompt. Altogether, the model was trained on the GPU cluster of the Kempner Institute at Harvard to produce the final model that has 11 billion model parameters, and both the final model weights, as well as training data sets and model evaluations are fully publicly released. There are a number of unique and interesting features that Procyon has, of which I would like to highlight 4 of them. So Procyon is naturally multimodal. As I mentioned before, the model can easily interleave multimodal inputs as well as outputs by combining text with protein sequences and structures. The model is not limited a priori to address only tasks that it has seen during training time. Instead, it can transfer to new tasks that are specified at test time. For protein function prediction, the important piece is that the model has unbound vocabulary of user input, so we are not limited anymore to predefined vocabularies and standardized ontologies for codifying protein function, and the model has both prediction as well as generative capabilities. So let me share with you a few vignettes of highlighting these capabilities of Procyon. So our first Our first work was extensive benchmarking of Procyon predictive capabilities. And here in particular, I'm showing just a snapshot of a subset of results, and many more are included in the paper that is linked from this slide, that shows zero-shot prediction performance of protein function across a broad range of functional categories. In extensive comparison against single modality models, such as protein language models only, structure-based models, as well as text-based models, as well as traditional models for protein function prediction based on homology detection and guilty by association principle, we see substantial lift in model performance measured by accuracy, Fmax score, as well as other established performance metrics. What we are most excited about is to see whether we can apply Procyon to new kinds of problems that would not be easily studied with traditional deep learning models just a few years ago. So here's an illustration of that, where we were interested in using Procyon to identify peptide sequences that are likely binders of ACE2 enzyme. So we started this application by taking the pre-trained Procyon model and training it on a small data set of naturally occurring protein peptide binders to produce Procyon bind. Once we had Procyon bind, we used Procyon binds to score and retrieve a set of non-naturally occurring peptides, such that those peptides that were scored favorably by Procyon bind were understood as most likely binders of ACE2. Our collaborators have been able to then synthesize and generate a large experimental data set of those peptides and measure the binding affinity between those peptides and ACE2. And we use that in data as information against which we then evaluated predicted peptide scores produced by Procyon. Importantly, because the data was experimentally generated, there is guaranteed that there is no information leakage from the pre-trained Procyon model to the data that was generated here. So once these peptides were synthesized and then binding affinity was measured against ACE2, we looked at the data and correlated predicted binding scores produced by Procyon against experimentally verified scores between these peptides, which were interestingly non-naturally occurring amino acid sequences, creating additional complexities for the model that had to overcome substantial distribution shifts. Nevertheless, it produced favorable results and scored highly those peptides that actually were bound by ACE2. In another application, we have worked with scientists studying Alzheimer's disease who are interested in understanding the phenotypic effects of mutations in key Alzheimer's genes. So here's an illustration of this study where Alzheimer's scientists started with PSEN2 and PSEN1 amino acid sequence, and then they presented to Procyon in their natural language prompt a wild-type sequence of a PSEN2 and asked Procyon to generate protein caption cards to predict what is the roles and functions that a wild-type PSEN2 sequence would have. But interestingly, what scientists have done next, they have started introducing mutations or variants in the wild-type PSEN2 sequence at locations where that are of interest because of likely pathogenic effects. And that has that mutated sequence provided as input to Procyon, then triggered Procyon predicting functions that likely mutant PSEN2 would have, but not PSEN1. A PSEN2 that would be wild type a protein that allowed us to start associating predicted functions with individual variants introduced into the amino acid sequence of the protein, and then overlaying those variants onto the predicted structure, linking genetic variants with protein structure and phenotypic effects that those variants might have. Finally, I'm showing here an example where we used Procyon generative capabilities to generate what we refer to as protein cards. So protein cards are generated functional descriptions of what the different proteins do that can be generated in a conditional manner by prompting Procyon to generate functional, disease, therapeutic, or molecular interaction phenotypes that proteins might be involved in. Shown on this slide is an example for AKNAD1, which is a gene whose proteins were recently implicated in Parkinson's disease. Reading this from left to right, we can see how human scientists provided to Procyon the sequence of this protein as well as predicted structure, as experimental structure is not yet determined. And so Procyon then generated a number of textual descriptions, which we call generated phenotypes, or just protein cards, that we were then able to validate using independent experimental data, including information from RNA-seq as well as the Human Protein Atlas and co-localization screens from there. Importantly, this is illustration of the protein that would otherwise would be challenging to predict functional annotations for, because of relatively low sequence similarity between AKNAD1 and other proteins that in SwissProt and UniProt that have some functional annotations. So it really highlights the power of Procyon by being able to explore vast amounts of biomedical literature as well as the instruction tuning dataset that Procyon was trained on in order to produce functional labels for this previously poorly characterized protein. So what we have looked so far is the ability of models, and Procyon in particular, to predict and generate phenotypic descriptions for individual proteins. Of course, proteins do not act independently of each other. Instead, they form protein complexes and form molecular interactions. So recently, Ada Feng, a PhD student in the group, has developed a model called Atomica. And Atomica is an all-atom model of molecular interactions that is specifically focused on modeling molecular interactions by modeling molecular interaction interfaces at the atom level between molecules that form molecular complex. Atomica is an all-atom model of molecular interactions. So instead of representing molecules in isolations, we want to represent molecules by generating powerful latent embedding space of molecular interaction interfaces of pairs of molecules and molecular complexes, which would allow us to better understand how molecules interact with each other in different biological contexts. The way Atomica operates is by starting from an interaction complex shown on the left part of the slide that we represent by an atom-level graph of that interaction complex, where we have nodes represent atoms in molecule 1, atoms in molecule 2, and then different types of edges indicating both intra and intermolecular interactions. And those atom-level representations give rise to block-level representations that correspond to common chemical motifs, amino acids, nucleotides, and other structures. Importantly, this is an all-atom-level modeling, meaning that the model is trained to capture atom-level chemistry, including bond lengths, dihedral angles, and other chemical properties at the individual atom-level resolution. Why is this important? This is important because an atom-level model allows us to test the model not only on dataset of proteins and protein-to-protein interactions, but also on dataset of other molecular modalities. Indeed, one of the key features of Atomica is that it's a first representation learning model that is trained across different kinds of molecular interaction modalities by being trained across small molecule to small molecule interactions, protein to ions interactions, interactions between nucleic acids and small molecules, between proteins and small molecules, between proteins and RNA, proteins in DNA, peptides, and proteins. So across the different molecular modalities and interactions that can form that span large amounts of physical scale in the atomic space, Atomica is a single unified model that can be trained in a principled manner because it's trained at the atom level, and those interactions persist across different molecular modalities. So when Atomica is trained, it produces a universal latent space of molecular interactions, and it's universal because we have a single unified latent space in which embeddings of interaction interfaces between different kinds of molecules are embedded. This creates many exciting opportunities for how we can use Atomica. I don't have time to show all of them, so I'll just show one recent example where we use Atomica to illuminate dark proteome. In particular, there are vast regions of the human proteome that contains many proteins that are currently poorly annotated. And so we wanted to use Atomica to better understand what do proteins in the dark proteome do. We used Atomica to predict 5 new heme binders, and those heme binders that were predicted by Atomica and by Ada were then later validated through an experimental campaign in the lab to confirm that those candidate heme binders that were previously in the dark proteome are indeed heme binders, and therefore we can functionally characterize them. And that was only possible by having a large universe and latent space of molecular interaction interfaces into which we were able to embed all of the proteins based on their atom and the other kinds of molecular modalities based on atom-level structural representations of molecular interaction interfaces. So we have so far seen two areas of scales of biology under which that AI scientist paradigms applies to molecular level reasoning and then molecular interaction level reasoning. Let's now proceed to understanding of how a biological context that those cells, that those interactions and molecules act in. So I'll start this part by a very quick motivation outside of biology. And so in natural language, we know that many words in natural language are polysemic words. For example, apple is a polysemic word. We can grow an apple, we can buy an apple. And to understand the meaning of the word in a specific natural language sequence, what we generally look at other words and sentence context in particular. And then the meaning of the word is resolved by the sentence context in which this word occurs. In biology, well, the meaning or the function of genes and their products and molecular interactions is resolved by biological environments in which those products act. H2FX is a pleiotropic gene, meaning that its the functions of its products are resolved via cellular or biological environment in which the products act. So the products of this gene in the environment with one specific drug would actually trigger and activate a different pathway than if this same gene and the product would be put in the environment with doxorubicin. So that obviously raises an interesting question, which is that of how can we develop AI models that would be more flexibly able to dynamically adjust their outputs to biological contexts in which they operate. And we have been asking and studying this question quite extensively by leveraging a large amount of single-cell AI datasets to build what we call contextual single-cell AI models, and then use contextual single-cell AI models to study disease and drug effects in single cells. So our paradigm to do that is to generally start by creating a contextual single-cell foundation model, which can be thought of as a general encoder of multi-omics or single-omics data that is disease agnostic and typically does not capture patient identity. So it can be easily shared across institutions and across different research groups. This is followed by stage 2 training of the model, where we learn an encoder of patients diagnosed with a specific disease in order to be able to produce patient-level representations and create a disease-specific foundation model that we can then, with little additional training and adaptation, use for a broad range of disease and specific fine-tuning or prompting tasks in order to enable cell-type-specific target discovery and target identification, cell-type-specific composite biomarkers identification, and treatment response signature modeling. We have so far applied this paradigm to inflammatory diseases, including rheumatoid arthritis and inflammatory bowel disease, have ongoing work in Parkinson's and rare genetic diseases, but I want to share with you in this talk today an illustration of this approach in the context of oncology and immunotherapies in particular. So I will highlight a recent model called COMPASS. And COMPASS is a model that is a generalizable AI model that can predict outcomes and in response to immunotherapies for patients, and can do so across cancers and across treatment types. COMPASS models, as we will see, can support a broad array of tasks by predicting both disease progression and modeling disease stage, examining genetic and molecular markers, predicting medication response, and help co-design disease treatment plans, and also characterize disease risk factors. The motivation for COMPASS was a realization that while immune check inhibitors have transformed cancer treatment and cancer therapy, the response rates of patients to this type of therapeutic modality remain relatively low and also variable. It varies across cancer types, it varies across disease populations, and it varies, response rates also vary across treatments. Furthermore, standard biomarkers such as tumor mutational burden and expression levels of PD-L1 and other key genes lack generalizability and often do not perform equally well as biomarkers of treatment response when we apply them to these diverse patient populations. So this creates a key important need, which is to have a predictor that would generalize easily across indications and treatments. The challenge, however, in developing large-scale foundation model style predictors is that we are limited here by relatively small number of patient populations in clinical studies, where we might have only hundreds, perhaps at most thousands of patients per each clinical study, of which only a small percentage of those patient cases in each study would respond to treatment. Which creates interesting challenge now. How can we train foundation models on data that is so scarce and poorly labeled and sparsely labeled? So this is a challenge that we addressed with COMPASS model. So COMPASS is a concept bottleneck model that is being trained to this two-stage training approach that I discussed just a few minutes ago. In step 1 of pre-training COMPASS model, we start with a large number of transcriptomes from cancer patients across several dozens of cancer types, with the goal of generating high-quality patient embeddings. And importantly, the unique feature of those patient embeddings is that patient embeddings are required to be described in the space of immunological concepts or immunological gene signatures, which is achieved by integrating concept bottleneck layers in the neural architecture of pre-trained COMPASS model. After step 1 of pre-training the model, in step 2, we can additionally fine-tune the model through supervised learning, or we can use the model in zero-shot manner. And we do that by leveraging small clinical cohorts and naive treatment transcriptome readouts of tumor microenvironments of patients from those cohorts. These then allow us to get a model that can generalize easily and is a large model while being able to capture the properties of clinical cohorts that would be relatively small in the number of patients that they would have. The model can then predict treatment outcomes that can inform precision medicine and precision treatments, trial optimization, and reverse translation. So how did we train the model through these 2 steps of training? So for pre-training, we used over 10,000 cancer tumors across 33 cancer types. And for concept bottleneck model, to design the bottleneck layer, we identified and developed immunological concepts based on over 120 curated gene signatures. Once the model has been pre-trained, of course, it can be fine-tuned through fine-tuning, partial fine-tuning, linear probing, or just used in zero-shot manner directly. And we test the model and these different fine-tuning modes across 16 clinical cohorts that are shown on this slide. Those clinical cohorts span a broad range of cancer types, immune check inhibitor targets, drugs, as well as vary from across very technical platforms in terms of biopsy site, sequencing technology that was used to generate transcriptomes, and the way the response rates of patients were decoded. For each of those 16 clinical cohorts, patient data are represented by the treatment-naive transcriptomes of tumor microenvironment, and then matched clinical metadata as well as response outcomes that are used for the model to evaluate its predictive performance. Let's look at some of the results of COMPASS model. So one of the first experiments we done was cross-cohort transfer, where we wanted to understand to what extent COMPASS model can generalize from one clinical cohort to another clinical cohort that it has not encountered during training or fine-tuning time. So the setting for this experiment was leave one cohort out setting, where during pre-training and fine-tuning stage, COMPASS models have seen the pre-training data set in 15 of the cohorts, and then at test time, the model has seen a cohort that was left out and never seen before. We can see here on the right part of the slide that COMPASS models outperform a variety of existing models, including standard biomarkers, as well as previously developed machine learning models, and do so across both large cohorts, medium, and small clinical cohort sizes. This suggests the model has robust performance across clinical studies of different different sizes and can generalize to a cohort of patients that it has never encountered before. Not only that, the model can generalize well across indications. So here we have designed a different data set split, where all of the cohorts from a specific indication, shown here as illustration, lung adenocarcinoma, were excluded from training and only encountered at test time. So for the case of lung adenocarcinoma excluded from training time, the model was able to predict patient response at over 76% accuracy, which was substantially better than that of strongest baselines in experiments. And this shows that the model can generalize well to new indication types despite differences in sequencing technology, biopsy site, as well as therapeutic properties. Not only that, the model can also be helpful to identify patients that would likely respond in early stage clinical trials, which is an important for downstream applications of patient selection or patient refinement and enrichment for early stage clinical trial design. And we have tested this capability of the model by emulating clinical trials and show how those clinical trials could be enriched by using COMPASS as a way to select patients and for early stage clinical trial. So finally, what I would like to show is that not only can COMPASS predict response to treatments for patients, in the sense that it can predict treatment response labels and associated probabilities, but what we can see is also that embeddings of patients generated by COMPASS are more strongly predictive of ultimate outcomes for those patients than outcomes that would be predicted for the same patients based on tumor mutational burden, PD-L1 expression levels, or established immune phenotypes, as we can see in this slide. The second important feature of COMPASS, which is due to its architectural design that relies on concept bottleneck layers, is the ability of concept to generate personalized patient maps. Shown here on this slide are two personalized patient maps. These maps can be thought of as human readable and interpretable prediction maps that COMPASS can generate and tailor to individual patients. So we can see here on the left an illustration of a personalized patient map for a patient who were put on PD-L1 therapy with tumor mutational burden of 13 and overall survival of 19.35 months. This is a patient whose tumor microenvironment was inflamed, and that patient indeed respond to this treatment. And we can see that in the activation map that COMPASS predicted likely response of this patient with very high certainty, and is then able to identify which immunological gene sets, the underlying individual genes, as well as their expression levels, the model relied on the most when making prediction. On the right, we see an interesting and somewhat surprising case of a patient. So in this patient was also put on the same therapy. They had higher tumor mutational burden, and their overall survival time was 1.44 months. Surprisingly, however, the tumor microenvironment of this patient was an immune desert. So we would not likely expect that this would be a strong responder to immunotherapies by conventional standards. Nevertheless, the patient COMPASS predicted that this patient would respond to this treatment, and indeed the patient did respond to the treatment. So by analyzing personalized patient maps across a large number of patients across in clinical studies, we were able to use COMPASS to identify and explore novel resistance mechanisms and be able to also perform stratification of patients in more precise granular immunophenotypes and show that those correlate with overall survival as well as response rates. So altogether, COMPASS combines and unites predictive performance with mechanistic interpretability to concept bottleneck layers. We show that it outperforms over 22 existing models for treatment response prediction across cancer types, across indications and therapies, and can support patient enrichment, early stage clinical trial design, and resist mechanism discovery. As such, COMPASS lays the foundation for more generalizable, interpretable AI models in precision immuno-oncology. In the last few minutes, I'll just share with you some of the very quick vignettes of our work at the fourth scale of biology, which is that of building and bringing together those various cell, molecular, and molecular interaction models in order to match patients to clinical outcomes across a large number of diseases and support the design of safe and effective therapeutic development in clinical setting. In this area, we have been developing agents, but not this type of agents that are shown here on this slide. Instead, we are developing AI agents. So I want to share with you a recent agent that we have released called TX Agent, which is the first of its kind reasoning agent that can reason across a universe of tools in order to support therapeutic reasoning. TX Agent can reason across all drugs developed since 1939 by leveraging the open FDA. It can reason over tens of thousands of phenotypes and disease phenotypes by relying on phenotypic descriptions from Monarch Initiative, and can perform real-time retrieval of knowledge and clinical insights from open targets. And this is possible by agent operationalizing and using tools to extract relevant information and perform relative analysis across all these types of data. TX Agent can establish a conversational model with human scientists, and it has three distinctive features. First is that it can perform open-ended multi-step inference and that inference is grounded on clinically validated knowledge, and the model performs cross-source validation by using tools and API requests to interact and query a broad range of validated resources. The model can perform multi-step reasoning, so it's not just a regular large language model, but really a reasoner that can handle more complex patient conditions, drug and disease information. And TX Agent performs well, comparably better than state-of-the-art reasoning models, including models that are orders of magnitude larger, as demonstrated by benchmarking done together with NVIDIA. The distinctive feature of TX Agent is that it's not just a regular large language model trained to next token prediction. Instead, the model is trained to use tools, and as such, it can perform autonomous analysis autonomously and use tools to retrieve and interpret relevant information to solve tasks at hand. So you can think about that in such a way that when TX Agent is presented with a question of task that it needs to solve by human user, it will engage in a multi-step inference process through which it will identify by relying on its tool rack system whether there are tools that can help answer the task that has to solve for human scientists, and then based on that tool that it can be retrieved from True Universe, send out API requests or perform different types of analysis to retrieve additional information in real time in order to then produce an open-ended answer for the human scientist to look at. TX Agent uses over 200 tools that are all trusted, tools from trusted verified resources that allow the TX Agent to perform real-time knowledge grounding and retrieval, meaning that even when the underlying data sources are updated, the model does not need to be retrained as long as the tool specifications remain the same. And that allowed the model to reason across all FDA-approved drugs since 1939 and associated validated clinical insights from other platforms. How is this possible? This is possible by relying on frontier in AI communication, which is that of leveraging model context protocols or agent-to-agent communication in order for the TX agent to then reach out to other external web-accessible data resources and tools and use them on the fly in real time to elicit its reasoning. And you can think of these protocols as the next generation of HTTP protocols that has really transformed how we think about today's internet. And so these MCP protocols allow us to also think about large language models in a much more richer, capable way. The challenge, however, it means that in order to train TX agent model, we need to train a reasoning model, not just a regular large language model, to the next token prediction objective. And as part of that, we developed a three-pronged TX agent dataset that we then use to train TX agent starting from a base LLM, and then develop a new benchmark that was used to test whether TX agent can reconstruct entire sections of FDA drug prescribing cards and can and benchmark the model on that set. So I'll just show one or two slides of that benchmarking process and then wrap up my presentation for today. So across different sections of FDA drug prescribing cards, we can see that TX agent outperforms in the ability to reason about the ingredients, warnings, dosage administration, pharmacology, toxicology, the use of drugs in specific patient populations, as well as clinical information of drugs, much more accurately than large language models, as well as other large language models that use tools and also use multi-step reasoning. So that was very exciting for us. Not only that, the model can take into consideration patient vignettes and patient history to produce personalized treatment recommendation. And importantly, because the model is grounded in knowledge, it retrieves through its use of tools and in external world. Each step of its open-ended multi-step reasoning process is grounded in validated clinical and scientific knowledge. This is the key feature that allows TX agent perform better than some of the larger reasoning models, where we compare the AP version of TX agent against DeepSeek R1 model, including the 671 billion model, and show substantial lift in performance of the accuracy across these benchmarks. TX agent is an open model that comes with open data sets, open evaluations, with a collection of Hugging Face models that I encourage you to check out, as well as an upcoming NeurIPS competition, CureBench, that we will release very soon. So with that, I will conclude this presentation. I shared a few vignettes of how AI scientist paradigm is transforming biological and biomedical research at the level of molecules, interactions, cells, and therapeutic development. And this would not be possible without a team of fantastic students who have developed and evaluated many of the models that I shared with you today. Thank you.
Thank you, Marinka, and very interesting talk. Maybe we have time for one or two questions. The first question is related to the initial part of your talk, Procyon. And the question is how to detect and avoid hallucinations in Procyon.
Yeah, so that's an important question. So that we have encountered ourselves, and the way we approached it is by relying on filtering of generated phenotypic descriptions by QA. So it is by question answering. So the idea really here was that when the model generated text that represents candidate phenotypic description or candidate protein function that the protein would have, then we take that phenotypic description and use it as input prompt and check whether the model would say, well, given this input prompt now, which was to generate phenotypic description, does the model think that the protein in question would likely have the answer yes or no? So this allows us then to take generated text and pass it back, perform multiple rounds of filtering through question answering with the same model. And we found that to be a fruitful strategy to filter out some of the responses that might likely be hallucinated. That allows us to decrease hallucination rate. However, the model still can hallucinate. It's just that with these additional mechanisms, we have better control over that hallucination rate. That is unlike the X agent, which in the multi-step inference process we require each step to be grounded in knowledge being retrieved via API requests to external databases, which then gives us some guarantees on hallucinations for the X agent, but not for Procyon.
Thank you. And maybe one last question about the other part of your talk, the COMPASS, the concept bottleneck work. So how many biomarker samples per subject need to be available at test time for good extrapolation and prediction of treatment responses?
Yeah, so for patients, in the information that we had across 16 clinical studies was mostly bulk transcriptomes of tumor microenvironment. So it was whole genome readout, but this was not single cell, but instead bulk transcriptomes. This was true for patient clinical data, which is big. And it's not surprising that here we have to have the model that can take as input not only single cell readouts, but bulk transcriptomes as well, because these type of readouts are still commonly used in clinical settings outside of basic biology data generation. So the model takes as input the whole 20,000, roughly 20,000 dimensional bulk transcriptome readout of tumor microenvironment from a specific biosite, which is treatment naive readout of the patient prior to the patient being put on treatment. And that is the only information that is necessary for the model to make prediction.
Thanks a lot. Thanks, Marinka, for joining us. I think it's probably very early in your time zone, but thanks a lot for joining us. And with that, we can continue with John Chodera, then our next speaker. Thanks, Marinka. Hello, John. Good to see you.
Hi, good to see you. Thanks so much for inviting me.
Thanks for joining us. So let me just start with a short introduction. So our next speaker, John Chodera, is a computational chemist and associate member at Sloan Kettering Institute for Cancer Research. His research focuses on developing quantitative models to predict a small molecule binding to biomolecular targets and understanding drug resistance. He's also the co-founder of Akira, where they build atomistic foundation simulation models to power the future of drug discovery. His lab aims to integrate physical modeling and structure-informed machine learning into cancer genomics and developing open source software for predicting drug properties. He's also co-founder of the Open Force Field Initiative and the COVID Moonshot, which shows his passion for open science and disseminating scientific best practices and also maximizing research reproducibility. And with this, we're looking forward to hearing your insight, John.
Thanks so much. It's a pleasure to be here. I've really enjoyed the talks today so far. I'm going to be able to tell you a little bit about some of the work our laboratory has done at the Memorial Sloan Kettering Cancer Center. I'm a part of the Basic Science Research Institute, the Sloan Kettering Institute. We're in the Upper East Side of Manhattan, right next to Weill Cornell Graduate School of Medical Sciences and Rockefeller University. We're always hiring every year, even still now, in the computational and systems biology program. So if you're interested in working in this really great biomedical research atmosphere, send us an application. We're hiring again this year. So our lab is very lucky to work with a lot of complementary technologies to advance the state of computer-aided drug discovery. A lot of that is modeling, and I'll tell you a lot more about that today. Some of it, which I'm not going to talk about too much today, is automation as well, where we can drive experiments in the laboratory in a way that generates the data that we need to build better models and validate them. We're also super lucky to collaborate with a wide variety of folks from different across the pharmaceutical industry or biotechs or other software developers that feed biotechs, as well as large data generators. I'll mention a little bit about this later today, and then very much collaborate broadly to build open source tooling and open data sets for powering the biomolecular research community. I'm going to talk to you mostly about drug discovery today and how slow and costly it is, something that you folks know very well, but part of the appreciation might be why it's particularly slow. And one thing I will note is that due to high failure rates later on in the process, especially during clinical phases, discovery itself, each program may not cost that much, but by and large, we're spending most of the money in overall aggregate cost per approved drug in the discovery phase, because we have to spin around these cycles so many times in order to even get to a molecule we can use to prove a clinical hypothesis or a disease hypothesis in some sort of efficacy experiment. Unfortunately, we as a society seem to be getting much worse over time at drug discovery instead of better. The cost per FDA-approved drug is now exceeding $5 billion in total research costs. That includes all of the failures, and it seems to be getting worse with time. So folks have coined this term Eroom's law to indicate the diminishing productivity, in contrast with Moore's law, of course, where we are able to cram more transistors onto silicon each time. And of course, you might ask, why is this? And the answer is drug discovery is really hard. Many of you already appreciate that. And I'm just going to talk a little bit about what I've learned about why that's the case and how we're working to address that. So as was mentioned earlier, I was part of this spontaneous global research collaboration called the COVID Moonshot, where we aim to generate a new oral antiviral against SARS-CoV-2 right from the beginning of the pandemic. It took us only 18 months to declare a new preclinical candidate for further development supported by the World Health Organization and Wellcome Trust by synthesizing from an early fragment screen. So this is structure-based drug discovery all the way to a very potent novel chemical matter. It turns out that Pfizer beat us because they had a drug that was almost an oral antiviral. It turned out to be IV ready, and they just made some tweaks to it. Fantastic molecule to get all the way through in just a year. But this speed of which we were able to progress this compound all the way from essentially a fragment screen to a preclinical candidate told us about how poor the current tooling is for actually supporting this kind of drug discovery. And just to give you a little bit more detail about how difficult this is, when you start a drug discovery program, you have to start with some statement of your objectives. So this target candidate profile captures what your objectives are for this multi-objective drug discovery program. It's sort of what properties your molecule has to have in order for somebody to be willing to give you a lot of money to develop it through the preclinical phase, which is fairly well regulated. It has to obviously hit the target, it has to get into cells and do the thing to the target. If you want it to be orally administered, it has to be reasonably soluble and then stick around the blood in a long enough time scale so that between doses, it's still hitting the concentrations you need. And then there's this whole panel of other ADMET-related issues or safety issues that you have to avoid. For example, you don't want to bind to hERG, otherwise you'll have adverse cardiac effects. So you have to incorporate all of these. And of course, this process currently is very iterative. It has very low success rates. It costs quite a bit of money. You have to make thousands of compounds generally. And it currently takes several years as you go through all of these stages to get to something that would be your lead compound that you nominate to succeed, to hit your target candidate profile. Of course, there's a lot of interest, especially with machine learning, to invert this problem and invert drug discovery in the sense that if I give you my objectives, maybe I can just conditionally generate molecules that satisfy this target candidate profile and then put them right into preclinical development. That would be great if we could only do it in 10 molecules. Of course, the challenge here is that data is not of the scale that we would hope for in drug discovery. The sum total of all public bioassay data, for example, in Kimmel is about 20 million measurements, and even the protein data bank only has a couple of 200,000 or 250,000 structures in them, only about 10% of which are protein small molecule complexes. This is about 6 orders of magnitude less than your favorite poetry generator, GPT-4, which is trained on about 13 trillion tokens, right? And so this is a 6 order of magnitude gap, where bridging that gap would require we send Sam Altman out to raise $6.2 quadrillion to scale up the current paradigm. Even that seems unlikely, so perhaps we need to be a bit more clever about how we approach things in the near term while we're missing this kind of data. So that means we're forced to do this iterative process for now, whereby we try to organize the assays that we do to meet this target candidate profile into a series of tiers so that we can fail early and fail cheaply. Of course, we still have to make the molecules, and so that compound synthesis might take anywhere from weeks to months sometimes if we have to switch the synthetic route that we're using. But a good chemist at Enamine, for example, can make a dozen analogs a week if we do things in the right way. And then we put them through various series of different binding assays and other kinds of ADMET assays early on, and then move on. If things pass that first tier criteria, we move on to the second tier, and then eventually onto animal tiers that are much costlier and time consuming as well. And hopefully we make it all the way to that last stage. Of course, most of the time we spin our wheels in this earlier stages trying to meet our tier 1 assay objectives. And then very few compounds make it all the way to the very end from this cascade. This process is something that you can actually simulate, and that's a useful thing to be able to interact with and think about how we would drive these with different strategies. We put together this little thing called Drug Gym, for example, where you can play with different stages of this cycle using oracles that are realistically trained on real data. In this case, we're just modeling the hit to lead part, where you only have about 3 objectives. You're trying to progress from an initial early hit from, say, a fragment screen all the way through stochastically making decisions like a med chemist would to hit your target candidate profile or lead candidate profile objectives in this case. So you can model this as a stochastic process. So what's cool about computer-aided drug discovery is that we can try to model each of these different assays that you might use in a real assay cascade with some sort of surrogate. And that surrogate might be something that's a ligand-based machine learning model, or possibly a mechanistic model, or the kind that I'll be talking about today are these physical models that rely on structure and some kind of physical interactions or simulations to produce a very robust, reliable, and generalizable prediction of what those assay readouts might be. And of course, structure-based drug discovery has really had an enormous time in the sun right now. There's now over 250,000 structures in the protein data bank. The last decade alone has produced 100,000 of those new structures. And of course, there's been a huge amount of excitement in your various alpha open bolts, Chi, Rosetta fold models that can extend this to proximal proteins in interesting ways to expand the universe of proteins that are tractable by structure-based drug discovery methods. Most of the time in lead optimization, we end up following a cycle that looks like this, where you have a current lead molecule that is maybe poorly potent, and you want to significantly improve its potency while not compromising any of the other properties, or maybe hold the potency constant while trying to improve the other properties. Generally, you'll have the chemists have worked out a synthetic route that allows you to install different building blocks or install different substituents using building blocks at different stages of the synthesis. And ideally, you want to make a lot of analogs at the last phase, because you can carry through a lot of the intermediate and then do a bunch of the final product synthesis in parallel, rather than starting at the beginning and carrying through all of these dozens of intermediates through, dozens of different parallel syntheses through. Now, the med chemists tend to make very conservative changes. These are trying to feel out the structure activity relationships of what this pocket will tolerate, for example, at this side. So these are very small changes, but in this reaction scheme with the building blocks that are, for example, in stock at Enamine, we can make any of 15,000 different molecules that are installing substituents at this R3 group. So you might wonder, what are we missing? What are the other things that we could possibly install there that might be a huge increase in potency that we hadn't considered from the traditional conservative medchem route? So we use these alchemical free energy calculations, which we spent many years developing in coordination with many other folks as well. What happens is that we're trying to compute the rigorous affinity change between 2 different compounds, or selectivity or other properties. And to do this, you run actual simulations at all of these intermediate stagings, where we interpolate one Hamiltonian to another that allows us to rigorously compute all of the statistical mechanical free energy difference that includes entropy and enthalpy and any conformational changes that are relevant as well. And that gives us a good, robust prediction of the affinity, but it's also something that we can use to predict a lot of other of the properties that are of interest in these target candidate profile assay cascades as well, including looking at selectivity, about whether it binds to one kinase or, say, a related kinase with a related active site. It turns out this is a little bit more accurate because you get fortuitous cancellation of errors. Also, things like changing atoms on the protein side to look at the possibility for resistance or broad spectrum. You can do things like optimizing thermostability for things like antibodies, or even computing a lot of ADMET-associated properties as we're getting more structures for ADMET-associated proteins like hERG and CYP3A4, looking at bacterial porin permeation, for example. So these are very generalizable techniques that we've put a lot of time into building open source tooling, and all of this tooling is now under the responsibility of the Open Molecular Software Foundation, where they're building a sustainable open source ecosystem for serving the community. So you can go to any of these projects like Open Force Field and Open Free Energy and download open source tooling that is minimally restrictive, MIT licensed, for trying out a lot of these methods yourself if you want to try to scale them up. They're computationally expensive, so I'll warn you about that compared to a lot of lightweight machine learning, but you can scale this all the way to production drug discovery, as we recently did with our ASAP AI-driven structure-enabled antiviral platform funded by $68 million from the NIH to prepare for future pandemics. So this ASAP Alchemy, for example, allows us to essentially do the entire structure-based drug discovery cycle to help prioritize which compounds we actually want to synthesize for our chemists at Enamine. And sometimes this works really well. From the COVID moonshot, we were able to actually make a lot of predictions that drove potency forward significantly in many different ways. The correlation with experiment often looked quite good, but you'll also run into cases that look like this, where you see that some subset of the data correlates very well with experiment, but in the end, you just make a bunch of predictions that had absolutely no correlation with experiment. And you can track down the details for things like this. And in this case, there's a protonation state effect that we were very improperly modeling in binding to the SARS-CoV-2 main viral protease for this series of compounds that's basically vertical. But this actually happens fairly commonly in different drug discovery programs. Christina Schindler of Merck KGaA, for example, did a large-scale survey of how well these methods worked in internal programs, and it turns out they work reasonably well about half the time, but about half the time you fail for various different reasons. Understanding these challenges is, of course, very important to progressing the field forward. I encourage you to read her paper if you're interested in the details. But also the other part is, even when they do work, of course, the accuracy is not what you would hope. There's a lot of different kinds of instances in which the accuracy is actually quite poor compared to what you would hope for in substituting for an experimental assay. And it turns out the more we can shift the inaccuracy towards more and more accurate predictions, the more the value increases superlinearly for these kinds of programs. Remember, we're going through many of these design, make, test, analyze cycles, and this here on is a log scale from our drug gym models. If you were able to run the experiment many times, you'd get a cumulative distribution function for how many molecules did you have to make, or how many design, make, test, analyze cycles did you have to go through, or how much money did you have to spend and time to actually hit your target candidate profile goals, or lead candidate profile in this case. And as you make the method systematically more accurate, it turns out eventually you speed things up enormously on this log scale, and the value per calculation in terms of how much you would be willing to pay to break even, it increases rapidly. So if we can make these even a little bit more accurate, it will save a huge amount of time and money and cost. So you might wonder, what is it that's holding us back in this area? Well, let's hop in our little time machine and go back to 1980s, in which Peter Coleman wrote down one of the first functional forms for the molecular force fields. These are molecular mechanics force fields that were used to model and run these simulations and model the interactions here. And what you'll find is that things have really not changed all that much since then. In fact, even if you use the latest kind of tooling from Schrodinger or other commercial vendors, it's essentially the same functional form, but with more parameters. What you'll see is that we've done the simplest possible thing in modeling bond stretching and angle bending. It is literally the lowest possible Taylor series that gives stable bonds, plus a very low order diagonal. So there's no couplings here, Fourier series that's truncated at n equals 6. And then there's a terrible approximation just because exponentials were slow on this DEC PDP-11, which is a late 1970s era machine that these things were designed for. This thing was designed for a machine that did 50,000 flops. That's 50,000 floating point operations per second, right? We have actual computers now that are literally 160 billion times more flops per dollar. And so it seems absolutely insane to me that we are doing things exactly the same way as we did in 1980. Surely we have a lot more flexibility we can get out of these things that can actually do lots of math. So let's think about how we can recast these things into a way that these modern computers can actually help us with. So if we think about how force fields are made, we insert graduate students and then turn the crank, and out comes parameter sets in which we hope that somebody actually uses these. If you look at things like the GAF force field, which is still pretty widely used today from the Ember community, this is the entire zoo of different atom types that somebody wrote down by hand. And if your chemical species have moieties that don't look like this, then you're going to have a very bad time. Plus, you can't actually extend these because the code that is used to parameterize these was never really released. And if you start to look at the things where you might have not just small molecules, but proteins, maybe that's binding to a nucleic acid and at some sort of interface, then you might need all of these different force fields that are meant to work together simultaneously. And you quickly realize that you're using work that adds up to 100 human years of hand parameterization over many, many years. This is not something that's readily scalable, and brings up the question of, well, maybe we can bring this into the modern machine learning area, where we actually just put compute to work instead. And so you might wonder, well, maybe we could take some of these wonderful abstractions that have evolved for machine learning, where we can construct a standard data set that somebody's curated, partition it appropriately into training and test sets, compile a series of abstractions in a way that allows us to chain them together in some meaningful manner to build a novel model, and then use best practices to train and evaluate this sort of thing. Wouldn't that be great if we could do this for force fields as well? These are the potentials that drive these simulations. And it turns out you can. This is one architecture. There's now several of these things that have come up in the literature where you can essentially treat this problem as learning atomic feature embeddings based upon the connectivity and identities of these different atoms in your molecules, whether it's a protein or a small molecule. You can take these embedded features that you've learned and then put them through appropriate symmetry preserving pooling to allow yourself to basically look up an interpolation from a different feed-forward neural network of different kinds of parameters, and then you could train it on whatever data you want. It could be energies or forces or physical property measurements, and then you can use a tool like Espeloma here on the right. This is an entire force field parameterization tool in order to actually fit these things and do it in a day. And what's cool about this is that your test set errors in trying to fit the quantum chemical data, for example, ends up being lower than the training set errors for more traditional typing methods like open force field or GAF or even the Amber force fields. And you can automate this process by doing it very quickly and fitting to various different kinds of molecules. You can even find that for some cases, molecules just are so poor in the traditional test sets that what happens is that humans haven't thought of the weird atom types that you encounter when you get to very large quantum chemical data sets. Simply, so some human has never written down that this weird nitrogen could be pyramidal instead of being flat, and that's why there's errors that are very large on traditional force fields. You could also show it things like peptides as well as small molecules and then put them all together and then parameterize the entire system, in this case except for solvent, in a way that is self-consistent and gives very stable simulations. And then you can start to work with very large data sets since this is scalable. This is the SPICE data set, for example. There's now other wonderful data sets like OMOL out there, where you've generated just a huge amount of data and have seen lots of different examples of biomolecular-like species and interactions, and then train a whole set of parameters. In this case, this is the Espeloma 0.3 set done by Ken Takaba here, and then do protein ligand-free energy calculations. Here, everything except for solvent is being modeled by the Espeloma model. In this case, we just don't have enough of the condensed phase data for solvent to be able to trust those parameters. So you can see it does actually quite well and has become something that's systematically improvable. And what's cool about making this systematically improvable is that we get to the point where we can start to think about leveraging the data we generate to systematically improve our models. What's wacky to me is that, where we are is as a field is that you'll make some predictions as a computational chemist, you'll synthesize some molecules, you get some new data back for the project. It shows that the model isn't very accurate, and then use the same model over and over again, hoping that things will improve in the future. There's a definition of insanity, for example, that it's the same, doing the same thing over and over again, but expecting different results. But we should be able to, using these learnable models, start to use that data to improve or fine-tune the predictions that we make by improving the model based upon the hard-fought data that we start to generate. So we're thinking about how we can introduce more learnability into these calculations. So you might think this is expensive, though, because you have to rerun simulations every time, and to take every step in parameter space, you might have to rerun simulations just to compute a parameter gradient, and that could take a very long time. So a student in the lab who is super bright, Dominic Rufa, now at Sandbox IQ, had this idea of maybe we could use a simple one-step perturbation where we say we're not going to run any new simulations. We're going to see how much further we can push things by taking several steps using reweighting ideas with some cleverness. And then he also thought that maybe if we reduce the dimensionality of the problem, it might be much better. So these Espeloma models, of course, have lots of parameters in them. Instead, he's going to focus on the simple electrostatic parameters, which are actually fed into a charge equilibration scheme, and think about making a low-rank approximation of what these atomic feature embeddings are in order to make just a small set of parameters that might be well regularized in terms of improving what these predictions might be for charges. He picked a simple example where he can compute rigorously what the free energies are of these of this transfer process instead of looking at protein ligand binding, which is a little bit hard to reliably converge. He's just looking at hydration free energies and explicit solvent, where we transfer a molecule from gas to solvent, and then looking at how we can shift the cumulative distribution of errors, where this would be like the 50th percentile. That's where the current errors are 1.3 kcals per mole. It's not actually 1.4 kcals per mole. It's not that bad, but it's something that potentially could be systematically improved. And the question is, how much can we do without any additional resimulation? So Dominic had this idea of using perturbation. So we'll use the samples we get from the current simulations at the current parameter set, and then try to minimize the error to experiment using this pseudo-Huber loss, reweighting the calculations on the fly to preserve the appropriate statistical mechanics. But the problem with this is that as soon as you start taking some steps in parameter space, you rapidly could lose configuration space overlap, because the probability density of conformation starts changing very rapidly. And so what happens is, if you monitor the effective sample size as you're reweighting, it can collapse almost immediately, so that you can't take very many steps before your predictions with this reweighted free energy become extremely unreliable. So what he decided to do is introduce some sort of regularization where you say, maybe I can tweak the direction in which I'm stepping by enforcing an effective sample size restraint, where once it starts to collapse, I'll gently push back so that my optimization direction parameter space moves me towards an area where I don't have to lose all that much sample size while still improving the objective. And it turns out this is surprisingly successful. If you look at the difference between these blue samples, where on the left, without regularization, you rapidly lose any phase space overlap and effective sample size, whereas if you let this regularization kick in, you can maintain it very nicely. And what this means is you can take many more steps in parameter space before you run out of this trust region, essentially, and are able to improve the overall accuracy substantially by almost a kcal per mole, even in this very simple system. So this is a very cool example where we can borrow some ideas about how to do this with low-rank approximations and very fast one-step fine-tuning, essentially, from things like large language models. But that hits an accuracy limit of how far we can go with these molecular mechanics force fields just by tweaking the parameters, right? Ideally, we'd like to go beyond the accuracy. There have been a number of ways for doing this in the past, for example, by introducing more of these cross-coupling terms. But of course, this is a weird basis to think in, because the number of terms you need to predict and the number of parameters explodes literally combinatorially as you get to more and more cross-coupling interactions. So maybe there's another way to learn these multi-body valence terms that doesn't involve this combinatorial explosion. And for our field, this was actually shown to us a few years ago now by Olis Isaev and Adrian Roykberg, where they introduced this new generation of machine learning potentials called Annie, where we encode the interactions between different atoms in a local region with these multi-body terms and push them through deep learning in order to learn the atomic contributions to the energies, which was able to fit DFT energies, quantum chemical energies, extremely reliably. And what's really cool about this field is that it's evolved at a ridiculously fast rate, where there's exploration of all manner of architectures of different kinds. We haven't really metastasized on transformers the way that LLMs have. And so there's a lot of exciting research in this area that's continuing to push things forward. And I continue looking forward to how it evolves rapidly over time. So it turns out you're able to use these potentials within the context of rigorous alchemical free energy calculations. In this case, we're just looking at what happens if we replace the intramolecular energetics of the ligand only. Remember, there's all these cross terms that are missing, with these multi-body terms that are learned from Annie potentials, for example. And you can show that already in some cases you can cut the error down in half simply by modeling the intramolecular energetics much better for these. And then you can expand this much further and then start to do free energy calculations using these neural network potentials in an accelerated manner. This is work with Gianni De Fabridis and the OpenMM team, showing that it really does improve the correlation to experiment more broadly on these benchmark data sets if we're able to use these Annie potentials intramolecular to replace the intramolecular energetics. these things. You spent some time making it easy for other people to use and play with by optimizing the kernels, for example, with this library called NNPOps. Working with NVIDIA, we're able to make this only about 3 times slower than GPU MD for molecular mechanics, which is amazing, but you do need to use a smaller time step to keep these stable. So it's about 3 to 6 times slower in this case. But one thing to note is that as these GPUs continue to get wider but not faster, the MM is not going to really get much faster because it can't really take advantage for the system sizes that are relevant for protein ligand interactions, whereas for these neural network potentials, they continue to use a lot of the GPUs, so they'll get faster and faster with each generation. You can now actually, in just a few lines of code, this thing called OpenMM ML, deploy these things yourself. It's all Conda installable, and you can choose between several different pre-built general organic molecular ML potentials to play with if you want to do it yourself. But I'll warn you that it's still very early days. You can see all sorts of horrors appearing in these simulations if you're not careful, simply because MD is able to access all of these weird, interesting conformational regions that weren't in the training set for a lot of these, including shooting protons off into space or having very different distributions of MM and ML torsions between these different potentials. But then you might wonder, do we really need molecular mechanics at all? By chaining together molecular mechanics and machine learning potentials, maybe I have the worst of both worlds rather than the best of both worlds. And so we've been working on ways in which we can just simulate the entire thing using neural network potentials. There's no bonds in these cases. It's all learned as well. And it's very slow at the moment, but it's something where a lot of the things that we took for granted are now much easier in this new potential where we don't have any singularities. We can engineer it to be the kind of behavior we want. In fact, it's actually really easy to do free energy calculations where you turn off one Hamiltonian and interpolate to another Hamiltonian. This is work that was done by Josh Fass and Marcus Feeder in terms of computing free energies that you wouldn't be able to compute with MM, where I literally break a bond and make another bond to compute the free energy between different tautomeric species in this case. And while this anti-potential wasn't really built for this kind of activity, and you see that the initial error is quite large, 7.5 kcals per mole, what's really cool is that you can fine-tune this model to experimental free energies. And as far as I know, this is the first time anybody has tuned a machine learning potential towards experimental free energies, but you're able to significantly improve it by several kcals per mole, in this case, eventually running out of steam, by simply fitting to some experimental data and then having a very small generalization gap between the training and test sets as you try to predict what the difference in tautomer free energies are for other species. So it's a cool indication that maybe we can not only simulate and compute free energies with full ML potentials, but maybe we can fine tune these things in the future. What's interesting is that, as I said, nobody likes this paradigm of iterating your way through all the way to target candidate profile objectives over many years, right? We would love to be able to do this inversion of the drug discovery paradigm. And you actually might want to ask yourself, to actually bridge this 20 million to 13 trillion number of labeled data points, how can we do that, right? And what's exciting here is that there may be the possibility, as we make these simulations more and more accurate to essentially approach experimental accuracy, we could eventually use that to label enough data to build a good emulator or fast ML surrogate model that allows us to label enough data points in some domain to actually make this generative modeling work in this area. And so this is super exciting. I think it's a very cool aspirational goal. It's something that is the motivation behind Ikaira, this new startup I've created. There's a number of wonderful folks that we've managed to get involved in this. The idea, we'll be building our first open foundation simulation model to share later this year, with the idea that we can iterate our way towards replacing experiments, at least at this first tier, with experimental accuracy. I'm excited to have something later to show you later this year, but I do want to talk about how we're engaged in a number of activities that are meant to try to make drug discovery easier for everyone, right? I ascribe to this idea that a rising tide really does lift all boats. And so there's ways in which we can collaborate in a pre-competitive manner that make things easier for everyone. And one of the most exciting parts of this is thinking about this really cool, fast work in this field of alpha, open, bolts, chai, Rosetta Fold, et cetera, especially with the bolts 2 debut of being able to predict not only protein ligand structures, but also affinities, which is something that's a very great way of thinking about how we combine this with more accurate simulations later on by providing initial starting structures, for example, and then crude estimates sets of affinities that can be used to prioritize which simulations happen later on. The challenge, of course, here is that there's only about 25,000 useful protein small molecule structures in the protein data bank. And if you go into Kimmel to look at the kinds of affinity data that's available, it's a bit terrifying because the correlation of one experiment with another is extremely poor. This is from a great paper from Sarina Rinneker and Greg Landrum, for example. What people may not have appreciated, though, is that the automation has become so good with X-ray structural crystallography that the new KO4 beamline, for example, at Diamond Light Source, which is the Siggatron that we used to generate all of the structures for our ACP antiviral consortium, which for a single project generated 6% of all new structures in the protein data bank since it began. It's enormously productive. Automation is so good that this new beamline will be able to produce one new protein ligand structural data set every 8 seconds. That's right, 8 seconds. It's 1 second to collect the data and 7 seconds for the robot to change out the sample. That's something like 50,000 structures per week. And if you think that we only have 25,000 structures in the PDB right now, you start to think, how can we use this resource to really change the amount of data we have and completely transform this field of protein small molecule structural prediction and affinity prediction? So we were able to launch this project just last week when I was in London, aiming to double the protein data bank protein ligand content in 18 months, as well as pair it with very high quality affinity data and then orders of magnitude more structure activity relationship data using nanoliter scale chemistry, and then scale this by orders of magnitude over the next few years. And this was super exciting. We were able to get together a large number of folks from industry for this roundtable at number 10 Downing Street. I sadly did not meet Larry the Cat, though that was my aspiration here. But you'll notice that, of course, in this benchmarking by Tristan Chodera, the accuracy of all of these methods falls off rapidly as we get further away from the region in which we've mapped very well. And so the first idea, of course, is eventually we want a model that has a phase transition and allows us to predict everything all right. But in the near term, we're going to be able to prioritize things, expansion of this structural data set space that is sort of steered by the industry partners joining this OpenBind consortium. So please tune in to openbind.ai and follow us for more information on this. I think it's going to be a super exciting project. One other thing I'd like to plug too is that the opposite of these targets, of course, is the anti-targets associated with ADMET. This absorption, distribution, metabolism, excretion, toxicity. It would be very impactful for drug discovery because 94% of the molecules that are made during lead optimization fail to meet ADMET objectives. So if you had a way of eliminating those, you can rapidly speed up the whole number of design, make, test, analyze cycles. And even then, a lot of these things fail during preclinical and then clinical phases because of ADMET issues. So if we had better models for this, especially structure-based models that allowed us to predict human genetic variation and that impact on these safety issues, we could get rid of a lot of these failures much earlier in discovery. Fortunately, there's a very small number of the usual suspects that are responsible for most of these ADMET issues. And so you might ask, what if I just had all of the structural data that was necessary to make predictions of how small molecules interact with all of these targets, as well as a ton of actual assay data that I could use to build and validate these models? That's exactly what this ARPA-H project funded by ARPA-H as well as Gates is doing with data generated by Octant and by structural data by UCSF, flowing through both curating data sets to deposit in various different places for folks to use in the ML community, but also running blind predictive challenges. We just had our first challenge in coordination with the Open ADMET and ASAP folks and Polaris Hub. It was really cool to see over 100 groups participating, so I hope to see you participating in future ones. And of course, all the data will be publicly available at the end of the blind challenge, deposited into places like Kimmel and the PDB, just as you would expect. A lot of this is under the aegis of the Open Molecular Software Foundation, so I encourage you to follow them. There are great folks trying to build the open source ecosystem on which we can have a thriving biomolecular simulation and drug discovery community. And I want to thank you all for your attention. I'm happy to take any questions that you might have.
Yeah, thanks a lot, John. Very insightful talk. And yeah, I think this is actually very interesting, and it might be different from the other talks that we had today, because you basically presented your insight about the molecular synthesis, which is basically before the clinical trials and the initial stages of drug discovery. So yeah, we have some questions. I will just read the questions. So the first one is, to which molecule size can this learned representation scale?
That's a great question. So the cool thing is that both the Espeloma like graph-based representations for predicting molecular mechanics parameters as well as these neural network potentials can scale to whatever you like, right? It will not do a good job if it hasn't seen things that resemble at least fragments of the molecules you're interested in the training sets, of course. But both Espeloma itself, for example, the charge predictions and the parameter predictions can work on very large biopolymers, parameterizing on the GPU in milliseconds. Neural network potentials, there's multiple different kinds. Some architectures are quite expensive, actually, on current generation GPUs, so that they take hundreds of milliseconds or possibly even seconds to perform an inference. But there's also a growing set of these things for which you can actually model hundreds of thousands of atoms, for example, for tens of milliseconds per step, or even less potentially, which would allow you to get competitive with current generation molecular mechanics simulations.
Thank you. And the next question again from Giovanni. Would this open bind project also aim to map intrinsically disordered proteins or proteins with disordered regions?
That's a great question. As far as I know, there is actually a new focused research organization in the UK funded by ARIA that's aiming to develop data sets that are useful for intrinsically disordered proteins. I can't for the life of me remember it right now, but I'll see if I can find that and put it in the chat later. I think we are very much focused on the specific set of things for which this technology is very well suited for high throughput. What that means is that we need high throughput soakable systems that robustly crystallize. Now, this is a pretty big constraint because you need a protein, of course, that has a well-defined structure, not too much disorder, and then good crystal packing while still having a solvent channel that allows access for a nanoliter droplet ejected by this acoustic droplet ejection echo to actually hit the top of the crystal and then soak the ligand into the binding site. But this does mean that you have the opportunity to start engineering existing proteins that may have some disorder in them to stabilize interesting conformations or interesting crystal contacts to make these robustly crystallizable. So David Baker and the Institute for Protein Design, for example, is part of this consortium with the idea of engineering the periphery of crystal contacts and possibly even engineering novel binders to encapsulate different kinds of interfaces that we haven't seen in these data sets so far, so that we can hopefully extend this to more structures. But of course, the AlphaFold-like models don't deal well with intrinsically disordered regions at the moment, right? It's very much, there's not a lot of training data, so it's very hard to be able to predict that. But it's exciting to see how this will grow in the future to deal with the kinds of detection technologies that may be informative for that.
Thank you. And so the next question is basically applies to also other machine learning models, predictive models. How do you quantify the uncertainty of these models?
That's a great question. So uncertainty is really important, especially if it's well calibrated in making decisions in drug discovery, because you'll want to make real decisions that have impact in terms of dollars and time and opportunity cost by making well-informed decisions as you're making these predictions. Traditionally, in molecular simulations, we've only had statistical error to use in making predictions, which is a great limitation because most of your error comes from the systematic error of having a force field that just hasn't seen examples of what it is you're looking at. And so I think one of the really cool things about these simulation models, that both on the Espeloma-like side for predicting parameters and then on the more neural network potential side for predicting energies more directly, is that we can start to think about training not just one, but an ensemble of these models that will allow us to propagate that after the fact. There's, much like I presented the single one-shot reweighting for extrapolating how we can optimize these things to fit some free energies, you can use the same technique to run a simulation at a central parameter set and then start to predict what that systematic error is, at least by perturbation with those parameter sets from the ensemble, either in the molecular mechanics or the machine learning potential regime afterwards. Now, this is not something that's been done quite a lot, but there's been some first steps in uncertainty quantification that the open force field effort has taken as well, that I think will lead to this really nice world where we'll be able to make much more reasonable predictions on a per prediction or per compound basis of what those uncertainties are.
Yep. And the next question is, how do you see the future of Akira? Are all releases going to be free and open source in future, and including for commercial use?
That's a great question. I can't speak to the future, what the future holds exactly, but I will say that there's a really neat kind of dividing line in our field that is between, in order to help someone discover a drug, the current IP system means that you need to have several inventive steps away from something that's in the public domain. So that means ultimately at some point one will need to generate some data that's in a held out domain in order to allow folks to patent molecules within that chemical space for which the model is most accurate. And that also means that the models derived from that may not be disclosable either, because you might be able to reverse engineer what data is in that particular domain. But anything that's built with public molecules that are already in public domain, or quantum chemical or experimental data derived from those, or anything that's exclusively built on that, there's no reason that the models or the code has to be proprietary, right? That this can be something that could be an open source public domain asset that the entire field can use to iterate. That's why I think projects like OpenBind and OpenAdMet are so useful as well, because we continue to produce more data into the public domain, as well as have an evergreen way of incorporating blind challenges into ensuring that we're remaining honest and not fooling ourselves about how accurate these things are actually prospectively. So I think there will be a healthy ecosystem of open tooling and open models that are relying on public domain data. And I'm excited to help continue to push for a generation of more public domain data.
Thanks a lot, John. It was great to have you in this workshop, and learning about your insights in molecular synthesis and all these open science efforts that you're making. Thanks a lot.
Thanks for having me.
And yes, with this we're right on time, and we have around 45 minutes break, and we continue at 3:30. And in the next section, we will have Jacob Kimmel, and then we will have Kexin Huang. Thanks a lot, and see you soon. Oh, sorry, 15 minutes break, sorry. Hello, Jacob.
Hey, Arash, how you doing?
Good, good. How are you?
Likewise, doing well. Appreciate y'all giving us the opportunity. I am gonna try to share real quick just to make sure that this actually works.
Thanks.
There we go. Can you see that?
Yes, I can see it.
Perfect.
But I think it's half of the screen. Or
Sorry?
could you share again? I think it filled. It didn't fill the whole screen.
Oh, strange. Okay.
You need to full screen. Yeah. Oh, it's small.
Is it advancing and everything?
Okay, no, okay. Okay. So we're starting 2 minutes.
Sounds good.
So hello again, everyone. Our next speaker is Jacob Kimmel. Jacob is the co-founder and CEO of NewLimit, a biotech company developing epigenetic for programming therapeutics. Previously, he led research at Calico as a principal investigator, focusing on aging biology, stem cell dynamics, and single-cell technologies. He earned his PhD from UCSF, where he studied how cell states evolve over time using time-lapse imaging. So it's great to have you here with us today, Jacob, and over to you.
Awesome. Well, thank you so much for the kind introduction and more so for the opportunity to share some of our work at NewLimit with all of you today. So as Arash just kindly introduced, my name is Jacob, and today I'll be sharing a bit of the story of NewLimit, a company my co-founder and I are building to try and add happy, healthy years to every human lifespan. So before we jump into some of the fun modeling problems that we've been working on that'll be of most interest to this audience, I want to try and give you just a bit of background on our overall goal so that it can motivate some of the work that we'll show a little bit later today. So in brief, we're trying to discover sets of transcription factors that can restore youthful function in aged cells, really taking the functions that you lose over time as you progress through your years of age and actually being able to reset those back to a more youthful state. We want to be able to do this by briefly expressing these transcription factors, and then we want to take those combinations of TFs, these payloads as we call them, and turn them into mRNA medicines that we can actually deliver to real patients, giving us a transient burst of transcription factor activity, but ideally a long-term restoration in cell state and function. And then we want to take those medicines and use them to treat the most common diseases of aging, the diseases that will affect all of us and certainly all of our families one day. So the science might sound a little bit far-fetched at first blush when you hear someone talk about restoring youthful function in an aged cell. But what I want to try and convince you of in just the next couple slides is that we actually already have some evidence that this is possible. And really, this goes back all the way to Shinya Yamanaka's work in 2006, 2007, later awarded the Nobel Prize in 2012. What Shinya and his group were able to show is that by overexpressing just 4 transcription factors in an old cell, say an old liver cell, you can actually reprogram that cell's type back to an embryonic stem cell. And alongside reprogramming its type, you simultaneously reprogram its age. If you actually take that young embryonic stem cell that you've generated and you differentiate it back out toward a number of somatic daughter fates, what has been discovered is that many of the features of aging are now abolished or ameliorated. And the extreme form, the extreme existence proof that it's possible to reprogram age is that you can actually take that young embryonic stem cell and give rise to a new animal with a whole life ahead of it, regardless of the age of the animal that you use to generate that initial embryonic stem cell. And now that latter experiment has actually been done on a loop. There's a group in Japan that did this 13 times in a row, taking a cell from an old animal, making a young animal, letting that animal get old, taking an old cell from it. And what I want you to take away from this prior art is that if cell age were something that were irreversible, like a law of physics, a continuous increase in entropy, then these results shouldn't be possible. And so we don't believe necessarily that pluripotent reprogramming as discovered by Yamanaka is actually going to be a medicine in and of itself, but we do think it demonstrates that many important features of aging can be reversed just by modifying the epigenome. And that you can do this by manipulating a fairly small number of target genes, a small enough number that you might even start to think about how you can drug them. Now, in addition to this existence proof of reprogramming cell type and cell age simultaneously, we know from other researchers that you can decouple these two phenomena, that you can reprogram cell type while keeping age the same. Groups like Marius Vernigs at Stanford and Fred Gage at the Salk have shown that you can take, for instance, an old skin cell and reprogram it into a neuron and retain many features of aging. So these two properties can be decoupled. Now then, the only particular axis on this coordinate system that remains to be explored is also, therapeutically speaking, the most valuable axis. We would love to be able to understand at New Limit if we can reprogram cell age while actually preserving cell type. This is really the opportunity that exists for therapeutics, where more often than not, you don't want to reprogram your old liver cells into neurons or some other cell type. You really just want to restore the youthful functions they've lost over time. So while it may sound a bit like a quixotic quest, when you actually reduce down to the pragmatics of the task we have to perform in order to be able to restore youthful function in old cells, it reduces down to a search problem of finding a combination of transcription factors that might evoke this mapping. Now, it then sounds simple. Let's just go out and find those transcription factors. But it turns out we're faced with a really arduous numeric problem. Even if you make some pretty reasonable simplifying assumptions, you're unfortunately left with about 10 to the 16 possible combinations of payloads that you might test. Now, this is about 10,000 times the number of stars in the Milky Way. It's far too many experiments for you to ever get in the laboratory and brute force your way through the problem. And so in addition to being able to run more of these reprogramming experiments than folks have in the past, it's also perhaps even more important that we be able to intelligently determine which of these experiments are even likely enough to work that they're worth doing in the real world in the first place. And that's really the modeling story that I'd love to tell you all today. Before we jump in, though, I want to start by trying to motivate as well what sort of outcomes we think are possible if you're able to solve this problem. And really, we think that the upper bound on the impact you could have on human health is really difficult to determine. It's quite massive. If you could restore youthful function, even just in hepatocytes, we think that could give you multiple years of healthy life, give you the energy and the metabolic function you had when you were in college. Likewise, if you could restore function, even just in T cells, we think that could go a meaningful way toward bending the curve of the increase in infectious disease that occurs as we all age from about 30 to about 70. And most recently, we've been working on trying to restore youthful function in the endothelium, the cells that line your vasculature and really form the highway network that allow all your cells and tissues to communicate. We imagine there are not only cardiorenal benefits, but likewise cognitive benefits that might emerge here. And so with that, I've hopefully shown you the majority of the cartoons you'll see today, and I want to jump into some of the data. I'm going to start by telling you a bit about the molecular systems we've developed, because I think understanding the data we're working with, the data we're able to generate, really gives you the grounding that's necessary to understand the modeling problem and where we've made progress there. So I'll break this down into just 4 easy parts, and we'll start off with the world of atoms and some of the experiments we've put together today. Now, given that intractably large search space, as any good drug discovery company must do, we've assembled for ourselves a drug discovery funnel that we refer to as our discovery engine. And this really consists of a series of technologies that allow us to narrow this massive hypothesis space down to a tractable number to test. Like most of those you all are probably familiar with, our funnel actually starts in silico, and I promise I'll get to that in just a moment. But today, what I'd love to tell you about is this interstitial period where we've developed some technologies that allow us to run thousands of these reprogramming experiments and measure their effect on cell age, all in a single dish or in a single animal. And these experiments really form the basis of the data corpus that we can then build models on top of. After we jump into those large-scale data generation experiments, I'll tell you a bit about the back end of this funnel as well, because ultimately what we really care about once we've discovered a given combination of transcription factors is not only whether or not we can make an old cell look like a young cell, meaning adopt a more youthful gene expression profile, a more youthful chromatin state, but if we can make an actual old cell act like a young cell. Can it actually perform the jobs it needs to perform therapeutically more effectively? So now talking about some of these phenotypic or looks like screens, as we colloquially refer to them, the experimental setup is actually simpler than you might expect. So all of these experiments at NewLimit start with groups of primary human cells. These are real cells from real human donors with the right number of chromosomes. They're not cell lines. This makes our lives much harder experimentally, but we hope makes the data commensurately more valuable. And we gather these cells from at least a dozen donors in each experiment. And we can introduce a pool of these transcription factors. And based on the randomness of life and the way we've designed the system, each one of these old cells will get some random combination of factors from within this pool, eventually spanning the entire space of possible opportunities up to a certain order, a certain number of TFs in each combination. We've then designed these tools so that not only can we demultiplex based on DNA barcoding and figure out which cell got which barcodes, but we can also turn them on transiently to mimic the dose that you would receive of reprogramming factors from an LNP mRNA medicine. It's actually not so useful to us to figure out what happens if you turn these TFs on and leave them on. That's not really the type of medicine we would want to make. And so engineering in this transience property made our lives a lot more difficult. But again, we hope on the back end makes the data more useful. We can then, as many of you are probably quite familiar with, actually capture measurements of what each one of these payloads does to an old cell state using single cell genomics. And then from those data, we can start to ask relatively simple questions. Did any of these payloads make an old cell adopt a gene expression profile more like a younger version of itself? So we run many of these experiments in vitro for T cells and endothelial cells. That's where this takes place. I'll also tell you just a little bit about one in vivo system we use to give you some grounding for the types of measurements that we're able to collect. So in addition to running some of these experiments in a dish, we've also developed some systems where we can run these screens in animals. So here's one example where for metabolism, when we look at human hepatocytes, we actually run these experiments in humanized livers. So the way this system works is that you have a mouse liver that has a genetic defect. So you can think of this like the mouse hepatocytes are sick. You can then complement that particular defect with a small molecule. And if you transplant in some human cells and then take away that drug, what happens over time is that those human cells are forced to regenerate. They're challenged to actually fix this damaged tissue. And what you end up with at the end of the day is a mouse that has a mostly human liver in it. Now, what we've been able to do is layer on top that same reprogramming chemistry I described. So now we can start to ask questions like, do any of these reprogramming factors make this old hepatocyte regenerate better, actually repopulate a larger fraction of this tissue, more like a young cell would? And we can likewise collect data here using not only those single cell genomics readouts, but we get a nice functional readout as well. How much did this cell actually contribute toward regenerating this damaged tissue? And now to sum up a lot of work in a single plot, we believe that these screens have led us to build a data corpus that's really larger than what most folks in this domain are able to work with. So in this embedding here on the left, each point there is actually one unique payload. They're colored here by some experimental batch variables just to show you that they integrate quite nicely without any sophisticated intermixing or conditioning. And just to compare to the scale of what's available in the public domain, to our knowledge, about 19 sets of transcription factors have been tested for their effect on cell age in all of the public disclosures combined. At New Limit, as of last evening, I think we've tested about 16,000. So we are several hundred-fold larger in terms of data scale than anyone else in this particular regime that we know of. Even comparing some of our data, these large single-cell perturbation data sets in primary cells, to the broader public domain, outside of reprogramming, outside of TF sets as your perturbations, we believe this data set is about an order of magnitude larger than anything else that exists in primary cells. And so for some of the modeling problems we'll talk about, in especially areas where we found success, I just want to emphasize that we think a lot of that success derives from the scale of the data corpus we're working with, as much from the cleverness of how we approach the modeling problem. So moving on now to actually talk about the land of bits, the raison d'etre for all of us to chat today. So in order to actually analyze these data and eventually build models that can run these sorts of experiments in silico, we first need to actually define what the measurements are that we're trying to optimize for. And so in one way of framing this, we have to learn to represent cell age from these complex gene expression readouts. When we start with a profile of 20,000 or so measured gene expression accounts, it's actually difficult in principle to go from that very complex profile down to a single scalar value that we care about. Did I make this old cell look like a younger version of itself? And so here, we've solved this problem by building discriminative probabilistic models using the age of a donor that a cell came from as a label. I like to think about this as the cheapest labeled data you're ever going to gather in humans. The assay for actually labeling each one of your cells is simply to ask the donor their birthday. It doesn't really get easier than that. And so we've actually accumulated a data corpus now of tens of millions of cells from hundreds of unique donors in this regime. And what we can do is train a discriminative model that, shown one of these profiles, predicts the age of the donor and gives you some distribution, some likelihood of how old the donor that cell came from might have been. Now, it turns out, as I'll hope to convince you in the next few slides, we can train these models in a quite performant regime. And then once we've built them, you can actually use them as something like in silico assays, a measuring stick for the properties we're interested in. If we, for instance, have two cells from the same old donor, we can predict the age of the control cell, and then we can show the model a cell that's been reprogrammed. Now, the model has no knowledge of that reprogramming intervention, but we can use the best guess of that from that model of how old that cell looks as a proxy for whether or not that reprogramming intervention has had the desired effect. Just to set the stage, we don't think these models need to be perfect. They don't need to have 100% discriminative performance. Rather, if they're actually even able to rank order things in the right order, we think that's actually sufficiently useful for us to then be able to narrow the field of hypotheses and start to identify some of those TF sets that are most interesting. So the way we've actually designed these models, again, is to train them on cells from individual donors and using those donors' ages as labels. And all the results I'll show you are from what we consider the most rigorous regime of evaluation here, which is a held-out donor scenario. We train the models on cells from some cohort of donors, and then we predict on another. And of course, you do that in your classic cross-validated fashion. And what we found is that we're actually able to build models that are quite performant here. This might be surprising to those of you who maybe haven't looked at data from young and aged patients before in this regime, haven't looked at single cell profiles from these sorts of donors. But it turns out that age is a dramatic perturbation, and it's actually not that challenging to achieve pretty strong predictive performance, given the right nuance and given the right conditioning. Now, not only do you actually get nice summary statistics, but I myself am always a fan in the modeling regime of qualitatively inspecting your outputs. Plot your logits. It'll save you a lot of time and help you find a lot of bugs. And here, we're happy to find that these logits are distributed in the way one might expect from a model of this kind. Predictions from young cells are mostly young, old cells are mostly old. And if we actually show the model middle-aged donors from an age it's never seen before, they correctly interpolate between those two extremes. Now, it's also important that the models we've built actually capture biology that's useful to us. One could imagine myriad ways to build performant models in this setting that might actually just be picking up on spurious correlations of one form or another, but don't actually reflect the underlying age of the donor. And so here, we've actually managed to set up a number of evaluations as well that ensure that we're not picking up on possible confounders. As one example, in T cells, there are many heterogeneous cell states. We know these cell states change in abundance over time in a way that is somewhat related to cell intrinsic aging, but not definitive as of such. And we found that our models actually have strong performance across all of these possible confounders. Likewise, for various experimental time points where you might imagine that the model actually gets confused or confounded. And finally, if you go in and actually use everyone's favorite attribution techniques, here we're just computing expected gradients, that if you actually compare those with the simplest possible prior one might have, something like differential expression of your features over time, here features being genes, they're actually incredibly strongly correlated, suggesting through various axes that these models are picking up on the real biology we care about, rather than some of the technical confounders that one might imagine in an experiment like this. Now, at the outset, I mentioned that not only do we want to reprogram a cell's age, but importantly, we want to preserve a cell's type. This is different than, for instance, what you're able to achieve with the Yamanaka factors. So in addition to being able to measure cell age, we need some quantitative way to measure a cell's identity as well. And we need those models to actually be quite sensitive. Even if a perturbation makes a, for instance, hepatocyte look still like a hepatocyte more so than anything else, but quite different than any physiological hepatocyte profile, out of distribution, even if still closest to the hepatocyte mode, that's probably less interesting for us because we know the target we're going after are simply young hepatocytes. They should be well within the support of the distribution of sampled cells we have access to. And so here, we've actually built a series of generative models that are based on a canonical VAE backbone. We've set these up in a conditioned way to integrate a number of different data sets, and the way we do that conditioning gets a little clever in how you set up your embeddings. And then once we've learned an integrated latent space, which I've shown you here on the right, here labeled with different experiments at New Limit, again, just to give you the qualitative sniff test that we're actually able to achieve quite a harmonized embedding here without any messy post hoc adjustments. This is just simple conditioning and training of VAE. We can actually fit a density estimator to those latents. And here, your density estimator can be as simple as a Gaussian mixture model. There's some priors here from the regularized autoencoder world that these actually work quite well. And then you can compute the likelihood of any cell profile under this regime. And so if we train a model like this across not only all of the cell profiles that we've observed from control cells, meaning not reprogrammed, but also the largest external data sets we can get access to that have hundreds of unique donors, again, tens of millions of cells, you can start to ask questions like, does this T cell or hepatocyte look anything like a T cell or hepatocyte someone has seen before? Does it look like a physiological cell? And if the answer to that question is no, then we want to deprioritize our investigations of that payload because the young states that we're trying to engender should be well within that distribution. So once you have these generative models and you can compute your likelihoods, you also need some way to measure whether these models are working or not. And this isn't an off-the-shelf problem where there's really established benchmarks. So we had to develop a couple different evals here to convince ourselves that these models were actually working. In the first of these evals, we've set up an in silico identity change, an in silico identity reprogramming event, where we actually just do linear compositions of our target cell type, the cell type of interest, call that hepatocytes, T cells, endothelial cells in our case, and then some other random cell type chosen from an atlas. Here I'm showing you random examples of kidney cells. And we ask whether or not the likelihoods of our model are sufficient to discriminate between cells that are 100% the identity that we want them to be versus those that have been linearly mixed with another cell identity. And we find here these models are actually quite performant. And we can likewise use our reprogramming intervention data as another evaluation. So we have some labels on top of those reprogramming payloads that we deliver. Some of those TF sets we know represent TFs from the native cell identity lineage. In the case of T cells, maybe as an example in this plot, those are lymphocyte TFs. Versus there are another number of other TFs that we actually include merely as controls that should come from a foreign lineage and therefore should be shifting a cell's identity outside of the normal distribution. And we likewise find that our models are able to discriminate between these two cases. So it's never perfect when you set up your evals, but we hope by taking a look at our models from a few different lenses like this, we can convince ourselves that the measures we're gathering are useful. And now I want to just try and convince you that cell age can be reprogrammed while preserving cell type. In some ways, this was the initial hypothesis, the initial scientific question of New Limit. And we now think we've been able to answer this in the resounding affirmative. What I'm showing you here are just a subsample of data from our corpus, and we're showing you the outcomes of those two models, the cell age model on the X, the identity model on the Y. And what you'll find here are that some of the existing controls, like the Yamanaka factors, do indeed make cells look younger, but just like we expect, they also abolish a cell's native identity. By contrast, many of the payloads that we've discovered at New Limit manage to reprogram a cell's age toward a more youthful state. For instance, we're able to take a cell from a donor who's roughly 80 and make them look roughly 40. There's some caveats around exactly how you do those measurements, but just to give you a rough order of magnitude of the sort of change in cell state here. And at the same time, we're able to preserve that cell's identity. A hepatocyte still looks like a hepatocyte, a T cell still looks like a T cell. In here, just showing you a zoomed in version of what some of those events look like relative to a positive control, which in this case is just a middle-aged donor you're able to shoot into the model that hasn't been reprogrammed, but just represents sort of a ground truth rejuvenation event. Now with that, we've actually managed to then build, using these models, a series of targets that we want to predict. If we were to build an ideal in silico model, we would be able to predict not only the effects of a given payload on cell age, but also identity a priori before we ever get in the laboratory. And then ideally, from those predictions, we could prioritize which experiments we actually do. And so that really forms the top of the funnel, revisiting our discovery engine here, where we're trying to build in silico reprogramming models, where these models should ideally take in TF sets as inputs and predict those targets. So in practice, the way we build these models is by setting up a conditional probabilistic setting, where we feed the model both a representation of cell state, some notion of a cell token, and some representation of the TF set, where for each TF, we have some embedding that we're able to learn, and we actually learn across those sets and then predict the conditional output using that probabilistic model. And we can set these models up to predict the whole gene expression vector that might occur as a result of a reprogramming experiment, the cell age, the cell identity. And now, once you have models like this, as is probably quite intuitive, you can start to do in silico screens, predict what will happen for a very broad range of TF sets, and then ask whether any of them look more exciting, meaning they reprogram cell age and preserve cell type, than others, and prioritize those experiments in the real world. So one question that we had to answer when we started building these models is how to actually represent transcription factors. And if you imagine trying to embark on this journey 10 years ago, we might have had to learn those representations from scratch, which probably would have taken much more data than we actually have today. Because we're now entering an era where we have foundational models of many of the molecular species we're working with, we can, by contrast, initialize our models with quite powerful representations that have been learned over these transcription factor molecules in other settings. So as an example, we can use protein language models like ESM, DNA language models like EVO to extract representations of these TFs and then use those to initialize our models. And here what I'm showing you is just an embedding of those representations that we've generated across a few different foundation models. And I've post hoc colored them with the families of the TFs that are well understood. All I really want you to take away is that the points that are colored the same way tend to be clustered together, showing that we're recapitulating some of the known biology we care about. And again, this is just a post hoc application of the label. These models have no access to it. We can then use these as our tokens as we start to train these in silico reprogramming models, as opposed to having to learn a new embedding with just a blank vocabulary starting out. So now, to actually convince you this works quite well, we've found that when we build models in this regime with our current data set, we're actually able to explain the majority of the variation in reprogramming effects that occurs in these single cell profiles before ever running an experiment in the laboratory. Not only is this true for individual outputs like I'm showing you here, but it's true across a range of benchmarks. And we'll actually be sharing some of these data at ICML in just a couple of weeks. This is also true across when compared to a number of baseline methods, and maybe we can talk in Q&A about what those methods are, almost any way that you think to measure it. The error of the gene expression prediction, the correlation of our cell age or identity predictions, the ability to top rank hits, really these models seem to be more performant than anything else we're able to build or anything else that's been reported elsewhere. And this is true also across the space of possible reprogramming outcomes. You could imagine another scenario where, for instance, the models we've built are really good at predicting one cluster of reprogramming perturbations. Here on the right, I'm showing you again an embedding where each point is a set of TFs and the effects it's had on a cell state. By contrast, what we found is that these models are actually quite good at predicting these effects across a range of diverse reprogramming biologies, and that in the vast majority of circumstances, the models that we've built here at New Limit are better than the best baselines that are available. Now, importantly, as I alluded to earlier, we think a lot of performance here actually derives from the scale of the data set we're working with. We've determined a pretty clear scaling law in these data where you get a roughly log linear improvement in performance as a function of the unique perturbations that you've collected. And so given that our data set is a couple orders of magnitude better than, or a couple orders of magnitude larger than what's available in the public domain, we imagine this contributes meaningfully toward the performance we're able to achieve. Importantly, I want to highlight as well that the scaling law really applies in terms of the number of unique perturbations that we've actually managed to measure. I like to think about this as we're trying to measure in some ways or estimate the density over the space of all possible cell states. And so really, if you think about collecting more samples, you want to collect as much support across that distribution as you can. Whereas collecting more and more cells from the exact same places within that distribution, we have found is actually much less useful. And so really, we think as we evaluate these data sets for perturbation prediction in single cell genomics, the number of perturbations is really the relevant criterion rather than the number of cells that you've collected. We then want to take these models and actually use them to accelerate discoveries in the laboratory. And I think this might be obvious as to how one goes about it, but we can then take these models and you can sample from the conjugate prior here. And by sampling from the conjugate prior of these probabilistic models, you can then design new reprogramming payloads. So rather than having to do simple, straightforward passes and ask of all the possible payloads that I can think to test, well, which of them has the best properties, you can actually define the properties you want and then draw samples from your model that try to optimize for those properties. So really framing this as a generative or design problem as opposed to just a discriminative framework. Now, in practice, one way you can evaluate how well this is working is by setting up what's called an active learning campaign. So the way that this works in general is that you have a series of experiments that are carried out over time, and you use these generative models to actually recommend which payloads you go test next. And in the ideal setting, if your model is working well, you're going to discover more of the interesting phenomena out there, in our case, hits in these screens, as a function of using these models than if you use some baseline, where here the baseline is effectively what we did in practice, the human guessing. And so what we found is that not only can these models beat the human baseline quite well, but that we're actually able to do this using a sampling procedure that's quite compute efficient. So rather than having to use a very expensive procedure like the upper confidence bound estimation, where we really have to sample through most of the possible search space, if you do the rough napkin math here, doing this across a very large experimental regime might cost $1 million, even in silico. But rather, we can actually use a simple technique like MCMC and cut that possible compute budget by 100 to 1,000-fold. We really haven't pushed the lower bound there, but just giving you a sense that you can do these types of design experiments in quite a compute efficient regime as well. So with that, I've shown you a bit of the data generation capability that we've built, a bit of the modeling that we've done on top of this. And really, all of this is in service of trying to nominate interesting payloads for therapeutics discovery. What we ultimately care about at the end of the day is not whether or not we can find a payload that makes an old cell adopt a young gene expression profile. It's whether we can find a payload that makes an old cell act like a young cell, treating a preclinical model of disease. And so what I want to show you next are just a few existence proofs that you actually can you can actually discover interesting payloads using these types of models, and that these types of payloads might actually have useful therapeutic properties. So I'll show you just two examples, one from T cells and one from hepatocytes. In T cells, we found that if you measure the ability of old T cells to actually kill target cells, this is one of their obligate functions in the body. When they see a cell presenting a foreign antigen, you're supposed to kill it, prevent that virus from replicating or prevent that cancer cell from growing, that old cells are actually substantially worse at this than young cells are. Now, this might seem obvious to you, or just prima facie would have been your null hypothesis, but it turns out that this is really the first time we think anyone's measured this in a rigorous way in humans. And we've likewise found that we can use these models to discover some payloads that are actually able to restore a youthful level of cytotoxic capacity in these cells. So you can take an old cell that doesn't kill very well, take one of these payloads that makes it look younger, and some of them actually make these cells act younger too. Now, if we jump over to hepatocytes, we've likewise found that we can discover payloads using these particular models that are able to restore youthful function in preclinical models of disease. And we're actually able to do this using LNP mRNA formulations, getting quite close to what an actual therapeutic would look like. I'll show you just a couple examples here. One is a model where we can injure a liver by effectively cutting it in half, and then you can deliver an LNP mRNA medicine and ask whether or not this helps the animals regenerate. It turns out when you do this with young and old animals, again, the old livers are quite poor at regenerating. They're about 2x worse than young animals might be, and this actually leads to substantial mortality in the aged animals, whereas all the young animals are fine. And that when we deliver some of the payloads we've discovered, we're able to restore that regenerative capacity back toward a youthful state, similar to the gene expression profile changes we observed at the screening level. Now, regenerating is not the only thing that hepatocytes do. Another really important property for all of us is the ability to tolerate alcohol. So it turns out that in aged animals, you actually experience substantially more damage from alcohol than young animals do. What I'm showing you here on the left is measurements of ALT and AST. These are just two enzymes that are expressed only in hepatocytes. The only thing you really need to know is that they shouldn't be in your blood at appreciable levels. And if they are in your blood, it's because hepatocytes in your liver are dying and leaking these enzymes into the circulation. So higher levels here are bad. These old animals experience so much more damage than young animals that you can actually just draw a line through the center of that plot, and you can tell an animal's age simply by how it responds to a drink of alcohol. Now, we've likewise found payloads that are able to restore a youthful resilience to that damage. And I find these data quite compelling on a molecular level, but the next piece of data I'm about to show you is actually one of the favorite pieces of data that I've ever generated throughout my career. And I think it's because it's so striking in a visceral sense, in being, rather than having these molecular abstractions you have to contend with, you can think about it in quite a concrete and physiological way. So another property that's different between young and aged animals is the sedative behavior they experience after a drink of alcohol. Many of you, like me, have probably run this experiment in your own lives. But as you age, the period of sedation you experience, meaning the period where these animals fall asleep and effectively have a hangover, is extended dramatically relative to young animals, shown you here in the center plot. And over on the right, what I'm showing you is that we found for some of our reprogramming payloads that restore youthful regenerative and resilience potential to hepatocytes, this sedative behavior is also rescued. It's one thing to look at it quantitatively like this, but I actually find it far more satisfying to just watch the raw data. So here's an example of young and aged animals that have recently had a drink of alcohol. What you'll see is that, like me, the old ones on the right are sound asleep. They're perfectly fine. They're just asleep. Whereas the young animals are kicking around, looking for their next meal, eager to go do something next. Now, here, what I'm showing you are two groups of old animals. All of the animals are old. The patches you see on their back are actually big swaths of gray hair. And the animals on the left previously received a reprogramming intervention before both groups were challenged with an alcohol diet. And what you'll see here, and I'll turn this sound off just so you don't hear the sound of our blower going. What you'll see are that the control, if this plays, what you'll see is that the control animals, sorry for the technical difficulties here. What you'll see are the control animals on the right are knocked out, whereas the animals that have been reprogrammed are just like their youthful counterparts running around. And so quite dramatic phenotypes, things like whether or not an animal is hungover as a function of an alcohol diet, can actually be reprogrammed using some of the payloads discovered using these models in the ensuing experimental system. And with that, I want to just close out here and thank all of you for your attention, thank our organizers most of all for giving us the opportunity to share some of this story with you, and to thank the team members who actually made this work possible. I'm the one from the firm who gets the opportunity to share it all with you, but really it was the folks on this slide who did all the hard experiments, who had the great ideas, and they deserve the credit for all the discoveries we've made. With that, happy to take any questions, and thank you all for your attention.
Thank you, Jacob. Very interesting talk. And yes, we indeed have some questions. The first question is, do we have a big, let's say, complete picture of what goes wrong in a cell when it ages in terms of DNA methylation, DNA damage, and things like this?
The short answer is no. We have not measured all of the different molecular species. And that's not only the we, meaning new limit, but I don't think we have a complete picture of all of the changes that occur with age. as a scientific community, there are always more molecular species we could be measuring. And just to give you a limit case extent of how many we could interrogate, there are things like non-traditional protein post-translational modifications that we don't even really have good tools to measure yet. So I don't think really the full painting has been completed. By contrast, what I do think is possible today is that using a few readouts that really integrate many of those other biologies in the context of, for instance, the epigenome, much of that epigenetic code is actually captured or integrated into the gene expression profile, which genes actually get used. And so I think we have a sufficient picture of what changes with age within a cell to begin trying to discover interventions. That's in some ways probably unsurprising. This is the premise of our firm to some degree. But I don't believe that it's comprehensive. We couldn't tell you molecule by molecule, locus by locus, every single change that occurs on the genome as a cell ages.
Cool. Okay, so the next question is whether these models, these age prediction models, should be conditioned on the tissue. For example, the skin tissues which are exposed to the environment may age differently than the other cells. So, do you think that there is a tissue-specific effect?
Yeah, great question. So we haven't looked at any settings yet where we're measuring cells that occupy multiple tissues or have been isolated from multiple tissues. But I think this is really the direction that these models will likely go over time, is being able to condition not only on a cell's type, which we do here as shown by this use of sort of a cell token, some representation of where did this cell start out in cell state space. But the more of these covariates you can effectively measure, including things like tissue, maybe even some notion of that cell's treatment or patient history as well, the more effective I imagine these models can become.
Great. And the next question is whether reprogramming one cell type is safe, or do we need to reprogram the whole organism?
Yeah, also another excellent question. We imagine that even reprogramming one cell type at a time is sufficient to provide meaningful health benefits. It's also much more tractable from a therapeutic perspective today. I imagine, based on the way that this underlying biology works, transcription factors have unique effects when they're turned on in different cell types because they bind different sites in the DNA. Effectively, when a TF is expressed, it runs around the genome and tries to find matching motifs in the DNA to bind and then exert its effects. Different cell types have different chromatin that's available. So even if I take TFX and express it in cell types A and cell type B, it's going to have two pretty divergent phenomena that emerge. And so based on that premise, we think it's unlikely you're going to find just a single payload you can deliver everywhere, which then brings you into this regime where you probably need unique medicines for different cell types within the body. And so what we've tried to start on, because it's going to be almost intractable to develop a medicine for various different cell types all simultaneously, is to focus on cell types where if you could restore youthful function, we imagine there'd be very meaningful benefit even from that one cell type alone. So I'll give you just one example in hepatocytes. There are actually human experiments in a way that have been done that measure just the effect of the age of your liver on the whole body. And this really comes from the regime of liver transplant, where you can look at aged liver recipients who receive donors either from old or from young patients. And you can ask then what occurs to the patients who receive young livers as opposed to old ones. And across most metrics one would think to measure, what you find is that even for physiological or organism level properties that you wouldn't necessarily think are tied to the liver, that having a younger liver is beneficial to these patients. And so we imagine that if you were able to just make the liver younger, you'd get a couple more years of healthy life.
Interesting. So even like making one tissue younger is generally beneficial to the receiver.
That's our contention as of today. I don't know whether that's true across the entire body. And so we've tried to be pretty judicious at New Limit with selecting the cell types that we work on so that we know if we're able to restore youthful function in this cell type, at least in some model system, that's sufficient to provide benefit. As just another example, one of the reasons that we work on T cells are that there are experiments where you can, for instance, adoptively transfer young T cells from a young animal into an old animal and then challenge them with something like an infection. And so the entire animal's old, except for a subset of T cells which are young. And it turns out that even just young T cells are sufficient to improve the responses to those sorts of outcomes. And so you don't necessarily need to believe that T cells are the only thing that matters. You really only need to believe that youthful ones would be sufficient to provide benefit to the patient. And so that's really the lens at which we try and view these problems. But we don't know if that's true for every cell type. We're only working on 3 right now, trying to increase our aperture over time. There probably are cell types in the body where that cell type isn't the weak link in some respective chain. And so even if you make it healthier, if someone else, some other cell type within some functional scheme is still broken, perhaps it doesn't help all that much.
Interesting. And the next question is, do you think these reprogramming effects are transient or long-lasting?
Great question. I wish I had longer term data to tell you about today, but as of right now, the latest we've looked is a few weeks after reprogramming. And just for context, in commercial LNP RNA medicines, these typically are dosed every few weeks by IV. So that's sort of the relevant time period for us as a minimum viable product. We found that even after a few weeks, these effects can be durable. Now, the real question is, how long can that durability actually last? Could these be something that you only dose every 6 months? Could they be something that you only dose every couple years? I think in principle, that's possible. And just to give you a couple data points on that, the most obvious is that epigenetic reprogramming is effectively how the different cell types in your body retain their unique identities. My eyeball very rarely accidentally becomes a pancreas. And so these epigenetic reprogramming events can be quite durable. There are other firms that have likewise done experiments now in non-human primates and some in cell culture over very long time periods where they've demonstrated in NHPs, you're actually able to get epi-reprogramming events to last years. That's just the longest anyone's looked. And in cell culture, a friend of mine, Luke Gilbert, now at the ARC Institute, once did an experiment where they passaged cells 400 times over the course of multiple years in an incubator after epi-reprogramming and found that the marks were still durable, even after simulating multiple lifespans worth of cell division and copying. So we think in principle, the upper bound on how long these things could last is quite high. We just haven't done those long-term experiments yet to demonstrate it with our own payloads.
Interesting. And maybe one last question. Do you use any foundation models like protein or DNA foundation models in your in silico experiments? And what is the biggest challenge in your method and also applying ML in this specific domain?
Yeah, absolutely. So one of the ways that we think we've managed to achieve reasonable performance to date is that we actually initialize our in silico reprogramming models from representations of transcription factors derived from foundation models of protein language and DNA language. And so by starting with this rich representation of each TF that already contains a lot of known biology in one sense, we think our models are able to be much more performant. And we've been able to demonstrate that with ablations where we try and learn this biology from scratch in our data set, and it just doesn't work as well. In terms of the biggest problem in our domain, I think this is a more general problem to reference, but I think this general domain of trying to predict the perturbation effects of genetic, chemical perturbations on single cell profiles is still really in its infancy. And I think the benchmarking historically hasn't been great, largely because the field hasn't really had enough data to work with. It's no one's fault in particular, but the data sets just haven't been large enough to build rigorous assessments. And so I think the most challenging piece for us has been along the way actually figuring out which evals we want to construct and which of them we actually trust to ensure we're moving in the right direction. I think that's often the case for modeling problems I've worked on in my career, is that the benchmarks and setting up the evals properly is harder and more important than necessarily trying to build the next fancy architecture or trying out that new training regime that you thought about. With that, thank you all so much for the great questions, and I'll turn it over to Kexin.
Thank you very much for presenting your work, and also looking forward to interesting results from Limit. Thank you. Hello, Kexin. So first, is this the right pronunciation for your name, Kexin?
Yeah. Kexin.
Okay, nice to meet you, Kexin.
Nice to meet you.
So our next speaker is Kexin Huang from Stanford University. I will shortly introduce you, Kexin, and then we'll be looking forward to your part. So Kexin Huang, a PhD student in computer science at Stanford University. His research focuses on leveraging AI for novel, deployable, and interpretable biomedical and therapeutics discovery, addressing challenges in multimodal modeling and uncertainty, and also experiment design. He also has done research in agentic reasoning in the space of biology and drug discovery. Kexin holds degrees from NYU and a master's degree from Harvard with extensive research experience in biomedical field. So with that, we are excited to hear your thoughts, and the stage is yours.
Cool. Thank you so much for having me, and it's really a great pleasure to be here. Yeah, let me share my screen. Okay. Can you guys see the screen?
Yes, I can see it.
Okay, amazing. Cool, amazing. Yeah. So today I'll talk about our recent work called Biomni, and this is our general kind of a research goal is like building a virtual AI biomedical scientist that can automate a lot of the biomedical research tasks. So first, I'll give around like 5 minutes of introduction of why we think this is a really important problem, and why is this a meaningful task, and then I'll delve into our project called Biomni, and I'll share about some results and demos for Biomni projects. So as you probably all know, biomedical research is fundamental. I think this is a great venue for drug discovery, and even moving beyond drug discovery, there's also like clinical diagnosis, agriculture, consumer health, cosmetics, and so on and so on. So for the past 2 years, we have been thinking a lot about what really prevents biomedical research from progressing. So people have been arguing, it's lack of ideas, but another major reason is also limited by human bandwidth. So what do we mean by that? When we talk to some PIs in the lab, they complain they have like 50% of data set out there. Just no one is looking at it because just like there's not much bioinformatician, there's not much computational people, and even not much biologists to dig into it. And when we talk to biologist friend, they all think, I have so many ideas. I have like 10 ideas, but at a single time, I can only do 1 to 2 ideas. So as we can see here, there's like a big deficit of demand and supply of biomedical expertise. And it's kind of this deficit is basically because of the limited of human bandwidth. And as the time goes by, I think the deficit is going larger and larger because as the new experimental technology has been progressed and there's new technology, new petabytes of new data has been generated. So to really close this gap, to close this deficit, we need a new way of scaling expertise. And one way to scaling it is just making more scientists. But we know that it is not possible, right? Because training a scale scientist is expensive, slow, and also each scientist right now is like getting a PhD is like very focused on one directions, and we must kind of fundamentally rethink how to scale kind of a biomedical expertise. So this motivates us to look at the AI agents. AI agent, what is it? In summary, is basically when human delegate a task to LLM, and the LLM can intelligently using this intelligence layer to decide if we want to perform action. And then an environment would act on these actions and then give some feedbacks about how this environment respond. And then this feedback just goes back to the LLM, and then the LLM can decide if it's finished answering, it can just return to the user. If it's not, just further performing the action and reasoning about it, and then so on. And we see that this simple paradigm has really helped the other domains. For example, for encoding, Cursor has honestly automated a lot of the software engineer stuff. As a PhD student in computer science, I actually don't need to run any code anymore, right? So it's like automatically scale this expertise. And then similarly for legal industry, there's Harvey, and so on. And we want to extend this paradigm to biomedicine because we think if we can scale this biomedical expertise, then just a lot of research progress can be exponentially done. So that is kind of the general idea that we have. That's can we, create a virtual AI biologist AI agent for biological scientists that automates biomedical research. So if we have that, then we can perform tasks much faster and potentially more accurately. And also, the amazing thing about automation is can tackle many of the tasks at the same time. And this really gives it much more new capabilities, right? Because previously, because of the limit of human bandwidth, a scientist can only look at maybe 10 genes or do a 50 gene screen. But now, if everything is automated, then there's no problem of selecting which genes. You can just do genome-wide because you can just run 20,000 agents at the same time. And then you can also have suggest and explore novel hypotheses and generate novel insight and discoveries as well. So we believe in this kind of a agent can really empower every scientist, every biologist. You know, in the future, a biologist will have an army of AI agent that's almost like research assistant that can get tasks to you, and each individual biologist, no matter you know how senior you're entry level PhD student, you can access to the top expertise as well. So similar effect as the cursor to software engineer, a biologist augmented by AI agent is equal to the impact of an entire specialist team. And it's also a great educational tool, as I will show later as with an example. It makes expertise universally accessible. It can dramatically reduce the barriers. And yeah, it just amplify and extend, and we believe it can push the research progress in totality, growing the knowledge of biology exponentially. So this talk is about how to build an AI biologist. To build an AI biologist, we first need to summarize how biology is kind of biological research is being performed. So we summarize into 2 steps. The first one is the generating hypothesis. So a biologist could use different, maybe explore some databases, reason about it, read some papers, and then gives an idea that, oh, this is an interesting hypothesis. This gene causes hair loss. And you can see that this step is very generic, but it involves many individual tasks and workflows. And then, given a hypothesis, you need to rigorously validate a hypothesis. And then this is where, for example, like computational biologist, biostatistician can look at all these hypothesis-free databases, like ENCODE, cell by gene, biobank, GTAX, and so on and so on. And it can write a lot of code, write statistical analysis, and to generate p-values. Or it could also be a wet lab scientist. It can perform a high-throughput screen to search for genes that regulate phenotype, and it can be like targeted wet experiment. For example, in this case, it conducts a CRISPR knockout for this gene, and it will observe the phenotypes of the mouse. So in summary, we believe bioresearch, in high level, is basically outer loop, is hypothesis generation, and then hypothesis validation, and then the validated knowledge will feed into refine a hypothesis, and then or just generate a new hypothesis, and so on. And within this outer loop, in each step, it involves a massive number of individual workflows. So basically, this naturally gives a recipe for building AI biologists, where we first need a generalist biomedical agent that can just perform a wide range of diverse, different biomedical tasks. And then building upon this general biomedical agent, we can then design specialized, maybe multi-agent agentic systems that kind of interleaves different generalist biological agent for hypothesis generation and hypothesis validation. So this is basically simulate how a biologist would perform the biomedical research. So our group has been focused a lot on this research direction around, on this vision, building AI biologists. So we have other works on agentic systems for generation and validations. But today we're going to focus on this fundamental part, which is building a generalist biomedical agent that can just perform a wide range of biomedical tasks. And this is work that we call BioOmni. So BioOmni is a joint work done with an amazing set of people, Serena, Hanchen, Jerry, Minta, and with our collaborators and also our advisors as well. As I briefly mentioned, now we kind of zoom in within each individual task. So within each individual task, there's usually a question, a task that is requested by a biologist. It could be, I want to run this analysis. I want to, I have this question. Can you help me do some literature research, and so on. So this very well-defined individual tasks. And then typically to get this task done, you involve multiple steps. It involves database queries, running some bioinformatics tools, search on the web, read some papers, reading about it, and even like performing wet experiments. And in total, it's basically a loop of reasoning plus actions, intrinsically speaking. And we know that while there's a lot of progress in reasonings, biomedical action space is just totally undefined. So what do we mean by this? So if we ask LLM, for example, ChatGPT, to really perform wet lab research, biomedical research, it just cannot get stuff done. Part of the reason is because there's no environment that enables the LLM to perform the actions and get feedback from these actions. So it cannot run some machine learning predictors. Of course, they cannot run wild experiments, cannot run database, bioinformatics tools, statistical analysis, and so on. So it's almost like a scientist without a hand. It just kind of keeps thinking and thinking. But we know that real biologists actually is a sequential step that just act something and wait for a bit and get result from their actions and then do reasoning again and then so on and so on. So this really motivated us that, you know, to really build a generalized biomedical agent, we first need to create an environment. Okay, then how do we create an environment? Because biomedical action space is actually really large, complex, and scattered. And we need a very unified, systematic way to create the action space. And we developed a new kind of method. We call this action discovery agent, which is a separate agent that we leverage the bioarchive. There's 25 subjects. In each subject, we can just get 100 recent published papers. And then within these 100 recent published papers, for each of them, we let another agent just read through each chunk of the paper. And then when reading the chunk of paper, we ask the agent would say what tools, what software, what databases that are necessary to reproduce the result in that chunk of paper. And then if it's and then we just screen it through this 2,500 papers across 25 subjects, and we collectively get a large set of tools, software, and databases that kind of systematically map the action space. So theoretically, if everything implemented correctly, everything implemented really accurately, we can basically the agent with enough reasoning abilities, it can potentially reproduce all of these papers. So, I mean, of course, it's theoretically speaking. And then now we have this kind of set of tools, software, and databases. Now the question is, we need to implement a lot of them, because these are basically we have the North Star, the entire action space, but it's not connected to the LLM. We need to have a method or have ways to create this environment that connect the LLM to the environment. So this is we'll introduce Biome E1. So E stands for environment. So for the first 6 months of the project, we're basically painstakingly a team of 5 people. We're just like working day and night, try to get this task, build this environment. Yeah, we initially tried to automatically generate it, but I think the result is not that ideal. But now, so we manually kind of create the environment. So we have went a lot of back and forth, and then we divided it into like 3 main categories that we think that are essential for the environment. The first thing is softwares. So these are like common biomedical softwares like ScanPy, P-Link, D62, GCTA, BioPython, BWA, and so on. And this software can either be like Python package, R package, or CLI tools. Yeah. And so for these softwares, we're not defining specific we're defining function calling, because we think these softwares are really popular, and the LLM actually has already seen a lot of examples in documentations on the internet. So it already knows how to write these kind of code. So we don't need to kind of write specialist predefined functions. It's just kind of like a limiting factors, right? It's because it's not that flexible. And then there's the 59 databases, because we know biomedical research databases are fundamental. This is where the real knowledge are coming from. And these include, for example, like Nomad, Open Target Genetics, RegulonDB, ClinVar, UCSC, Interpro, AlphaFoldDB, and so on and so on. And then for these databases, we make it super simple. So it's just one agent. We call this database sub-agent that it will, given a natural language query, it will automatically write a piece of code given the database schema, and then retrieve the relevant data corresponding to the natural language queries. And then so that's the 2 main part. And another thing is that we also find there's a lot of specialist knowledge that are not covered anywhere on the internet. These are like knowledges hidden in the biologist's brain. And for these, they're essential, right? Because if we want the agent to have like expert level performance, we need to have these specialist know-hows that are kind of distilled to the agent. So these include wet lab protocols, for example, golden gate cloning, oligo assembly, parameter design, and so on. And another part of specialist tool is the AI models, because if you ask LLM to generate a complete script to run an AI model, right now, especially on these like really new AI models, it just cannot do it. So we also provide some functions like pre-specialized tools that can perform AI models like diffdoc, UCE, and so on. And then yeah, of course, there's a lot of know-how, like other kinds of know-hows as well. So together we have this kind of an environment, we call it E1, which is a single environment with diverse tools, software, and databases. Cool. And now we have the environment. Then the challenge is, how do we utilize the environment? How do we have the agent to utilize the environment really well? And we propose a new agent architecture, which is called Biomni A1. A stands for agent. So the first step is retrieval, because our environment is pretty massive. It can hundreds of tools and hundreds of software and databases. And we know that to answer a question, it's not we don't need everything. We only need a very few tools and few database and softwares. And this entire without retrieval, it could just put everything. It just utilize a large number of tokens that's kind of unnecessary. So that is why we need a retrieval step to retrieve the relevant resources to answer these questions. And we experiment on different retrieval techniques, like embedding-based approach and so on. And in the end, we just use almost like a sub-agent, with kind of doing the retrieval, like tool retrieval for the task. And the part reason is that because we find that to really precisely identify what is relevant tools, software, and databases, it actually requires some reasoning ability as well. So the embedding-based approach is just like the similarity-based approach. It's just not ideal. So after we have the retrieval, then the natural question is, I have this list of resources. I have these questions. How do we go about it? How do I address it? So our motivation is like, because we want to build a general purpose biomedical AI agent. We don't want to predefine a gazillions of different tasks, like this pre-specified workflows, which is most kind of the current work they're trying to focus on, that are very specialized for individual tasks. It's kind of infeasible, right? Because we have just so much biomedical task out there, and each user will input slightly different words, like different sentences, and there's a lot of subtleties in it. But we know that LLM actually, in the knowledge it has read, millions of papers, it probably read all the forums, all the dialogues. So it knows a lot about how to perform a task, make the plan. So here we're actually just relying on LLM to automatically create a plan based on these resources and the input task. And this planning step is also adaptively tracked. And in the future, once there's maybe some errors, it will basically update the plan and adaptively. And then also, if it senses if it senses a step that kind of requires a change of plan, it will also change the plan as well. Cool. So that's the planning. Then, given each step of the plan, we need to really perform the actions. We need to get this task done. So traditionally, people use function callings. Just define functions, and then the LLM during pre-training, post-training, it is kind of fine-tuned to use these tools really well. But in our environment, it's actually pretty specialized, right? For example, we have softwares, and for these softwares, there's no predefined functions. Similarly, we have a lot of raw data sets, and also these pre-specified tools. So the natural question is, how do we intelligently use everything all at once? And the function calling approach just does not work naturally. And it's also just pretty redundant in some sense. There's like many steps, and then we use kind of a coding as a way of replacement to perform the actions. Basically, what coding does is we basically teach let LLM know that you have an environment installed with this list of softwares. You have these data sets, and then you have this specialist tools, and then you need to write code to answer each step of the questions. And the coding is really great for this. One benefit is of course it's kind of compatible with our environment, and the second thing is it just requires much fewer actions, right? For example, for the rare disease diagnosis task, giving an input a list of HPO terms. For function calling, just basically go through it needs to call like 6 times, but for coding it just needs to write a for loop. So as you see here, coding is a very effective way to generate task solving, and it's really fit for purpose for our environment as well. Cool. So that's an overview of our agent. We actually have experimented gazillions of different variants of this agent, but the simple and less structured works the best. So given the input question, we have the tool retrieval, we perform the reasonings, and we use coding to for compose actions, and an environment will execute this coding, and then you will return some of the observations, and then you will do the reasoning again, and so on and so on, until it arrives at an answer. Cool. So that's kind of the method, and then we also spend around 6 months on trying to rigorously evaluate and study how is this really useful. So we first use a Future House exam style benchmarks. So we choose 2 subtracks of the Future House lab bench because they are too kind of dependent or environment dependent. So the first one is around database query, and second one is the DNA sequence manipulations. And we first use a development set around 45 questions from these datasets, just tune the agent, tune the datasets, and stuff like that. And then we tested on a holdout test set, and then we were able to see that Biomni has the best performance, similar to humans. And we can see that, the baseline here is the LLM, which is what most biologists are using right now, and also the React, which is the agent that just can interleave a lot of LLM calls without much tools. And then this is the coding agent, which is the agent equipped with coding tools. And then this is the literature research agent, and then coding plus literature research. And this is the React, which is ablation of the Biomni, which does not use coding, but it actually uses function calling. And we are able to see that it has pretty good performance, but we know that this is not enough, right? Because this is more like sanity check, since we kind of tuned on like with the development set. So we tested on a holdout. We never seen before exam style questions, which is humanities last exam. There's no development set in this, and we're able to see that Biomni has pretty great performance compared to especially the LLM, which is what biologists are using right now. And this humanity last exam are like really tough questions, right? This is like by the name, it's the last exam for the humanities, right? So we're able to see it has 17% accuracy, and the LLM has like 60% accuracy. Cool. And then after that, we see that there's some signals, but we know that this is still not enough, because we're interested in let the agent to really perform real-world biomedical research tasks, just not some exam. So we created 8 new tasks, new benchmarks that actually measures the performance of the LLM agent in the real world. So we have, for example, causal gene detections, which basically have a GWAS hit for this trait XX, and you can look at the neighborhood genes around the GWAS loci, and want to ask which is the causal gene. And for the perturbation screen design, given a cellular context here, T-cell exhaustion, we want to develop a screen of CRISPR screen of 50 genes, which genes to pick. And then we have a kind of some ground truth to measure that, which is basically for some genome-wide experiments, we can measure whatever genes you choose. And then for single-cell RNA-seq annotation, we basically have single-cell RNA-seq data start at this path. I want to annotate a cell type for me. So for each cell, I want to generate a natural language cell type, and we're using some semantic accuracy, LLM accuracy, because the LLM is like kind of flexible to generate whatever cell type name for the for each cell. So we can only use semantic accuracy compared to the author annotations. And then similarly for the microbiome disease taxa, for which this data set in microbiome, we ask about expert bioinformatician to perform the same task to identify the taxa that are associated with the disease. And then we have an agent to also run the same task. And for the drug repurposing, is I have a rare disease, and just among a list of potential drugs, we want to find the most likely one for repurposing. And then for the last two, we have rare disease diagnosis, which is I have a patient with phenotype, like a list of HPO terms, and the whole genome sequencing returns a list of gene mutations. I want to know what is the diagnosis. And this is a variant of the task. Instead of the diagnosis, I want to know what is the causal gene. So overall, you can see that this across all the benchmarks, we compare with the LM, we compare with the coding agent, and we also compare with the Biomini React, which is an ablation of the Biomini. And we're able to see that at least compared to LM, it actually has more than, I think, on average, more than 400% improvement. And also for React coding agent, it has more than 40% improvement. And the React, it has more than 10% of improvement. So I think overall, we see that Biomini is a kind of strong agent that can perform just a wide range of different research tasks. And also just to see that this is a completely zero shot. We actually just only tried one pass because it's just pretty expensive. So we only have budget to run it once. So it's like a YOLO run. And then we're pretty amazed to see that across all the tasks, Biomini has the best performance. Cool. And even after that, we're still not satisfied. We want to really let the biologist to feel and touch it, and then to use it. So here we're collaborating with Le Cong's lab. So Le Cong is a CRISPR pioneer who have done a lot of functional genomic screens in his lab. And one of the students, is basically they want to look at a tedious but a very important task, which is cloning, which is fundamental for many wet lab experiment research. So for this task, it's like I have a plasmid sequence here, which is very long, and I hope to clone a CRISPR-guided RNA targeting human B2M genes. And can you generate a final map of the plasmid and also step-by-step guidance? And then Biomini is basically given this input question, it performs some analysis, it runs some tools on guided RNA design, it have article design, article annealing, and so on and so on. And it takes around 8 minutes to complete. And in the end, it generates a protocol. So for this protocol, we're actually, originally, we're actually just using LM to hallucinate it, but it just not perform really good. So we're actually just borrowing the protocol from Le Cong's lab. And in the protocol, we have a lot of missing parameters. These are, for example, the guide RNA sequence and some parameters here. And then these will be generated from other tools that are used before this protocol function is called. So we think after this one, then the protocol is like really robust, because this is basically how human scientists are also doing it. For this standard research task on protocol, people don't need to rewrite all the protocol. They just need to have a few free parameters, and then you will use a lot of different tools to fill in these kind of parameters. Of course, for more advanced cloning, for more advanced wet lab protocols, then we will need to kind of generate the protocol from scratch. And for that, we're actually working on that right now. We can utilize basically interleave multiple different protocols to generate new protocols, and this also gives a kind of more factual groundings to generate when you generate the new protocols. Anyway, so after we have this protocol, a scientist basically follows the protocol to perform the cloning, and we're able to see that the cloning is pretty successful. If you see all the red dots here, and then the sequence is also aligned with the AI generated sequence. And the scientist Jerry is very happy. He says, cloning is hard. It takes 3 years of training, and even then, biologists spend hours navigating tools like SnapGene, AlloGene, and primer blast just to design one construct. And Biomini can do it, within 10 minutes. And then since then, we also, really want to understand how impactful is this. Like, we want to evaluate it more systematically. So for cloning, there's many different scenarios. So Jerry come up with 10 real scenarios that he encountered during his wildlab experiments. And on these 10 open answer cloning scenarios, Biomni had similar accuracy as expert and greatly better than trainees. So what we mean by this is we have like 4 kind of categories. First is LM, then Biomni. Then we hire a trainee, which is like a master level Stanford student who have actually done cloning before. But she's not an expert in cloning. And for expert, he's the one who has his postdoc at Le Tong's lab, has done cloning for like 8 years. It's like a true expert in wild lab experiments. And then after we ask each of these 4 categories, answer all of these questions, we have another human expert to blindly review and to give grade to each of the categories. And we're able to see that Biomni has pretty similar accuracy to the expert, and then it has a big improvement on trainees, and of course a big improvement on LM. LM is just going to hallucinate all the ways. So this can really give evidence that we can actually achieve expert level performance with the right tool, with the right environment, and with the current reasoning abilities. And I think this gap between the trainee and Biomni is really interesting because this basically shows that this is a great educational tool. Because previously for a trainee to learn how to do cloning, you will need to bug, I don't know, the senior PhD student for like 3 years just to learn everything. But now you can just bug a Biomni, to the agent to teach you how to perform this, how to perform that, and only when there's like a really tough question, you can ask the senior members in the lab. So this makes the senior labber more effective and focus on for their own research as well. Well, so that's the wet lab analysis. And then we also think this dry lab analysis is also where the agent is gonna shine. So we first we work with Michael Snyder's lab. So Michael Snyder has a lot of this wearable sensor data, and for these wearable sensors, the data are often like really complex, like because they're all raw Excel sheets, massive number of them, and it actually takes many times for bioinformatician to analyze, to pre-process and analyze this data. So Minta, who is the postdoc at Michael Snyder lab, was working on this data set right before we asked her to do it. And then she actually finished this data analysis on this data set, and it took her around several weeks, like 3 weeks, working 2 to 3 hours per day to complete. And then basically for the same task, we just asked the agent Biomni, okay, I have this folder which contains 458 raw Excel sheets of continuous glucose monitoring. Can you pre-process data set, analyze data set, and generate some meaningful hypothesis? And we are able to see the Biomni in like 35 minutes, just pretty autonomously without human interventions. It just perform data set exploration, pre-processing, event detection and temporary analysis, and so on, and generate gazillions of figures, visualizations, and generate some interesting conclusions. And interestingly, this conclusion is also what Binta has able to find when she was doing the analysis before. So you can see that this is a dramatic improvement on the productivity, right? Because previously it takes 3 weeks, now it's 35 minutes, and it's like 100 times less times. And we are also pretty amazed because right now you can actually parallelize it. You can run like 10 different tasks at the same time, and this is just like 100x, even 1000x productivity boost for biologists. And so that's kind of on the wearable physiological biological science. And now we're also looking at like multi-omics analysis, which is more kind of expert level, requires a lot of specialization, requires a lot of know-how. So Han Chen there is an expert in single cell analysis, and he was looking at a database, a new data set. It's a single cell RNA and a single cell ATAC-seq data set. And then he basically curated a very long prompt, a very detailed step-by-step instruction on how he wants the agent to perform the tasks. So because this requires very specialized knowledge, we find it really beneficial for the human to just put a very detailed prompt so that the agent can understand it much better and follow the instruction and perform the task for you. And so for basically perform all these different steps, you run in Pythonic, on a GI boost to AU cell, attack, and so on, and generate a lot of visualization as well. And in the end, it generates a novel transcription factors that it identifies some interesting hypotheses. It also kind of recapitulates some known hypotheses that are reported in the original paper. But it can also, like after reanalyzing the data set, it generates some novel transcription factors like AUTS2, ZFHKS3 that showed high regulative activities just across multiple skeletal lineages. And we asked this Jin Tian and Peng, who are like biologists who are expert in multi-omics analysis, to manually examine the trace of the agent, look at the output, and they all kind of validate these steps. Cool. And so this is also only just like a tip of an iceberg of what BioOmni can do. So since then, we are trying to just have fun with it and then experiment on different tasks and working with different labs on what kind of capabilities can BioOmni do. For example, it can do like variant interpretations. Like given I have a GWAS loci, I have this disease, can you give me a genetic mechanism for that variant? Can you interpret the variants? And you want also to do on the more drug discovery applications like ADMED property predictions. Given a SMELI string, I want to predict the ADMED profiles, so some drug likeness profiles. And I also like just want to download some data sets on the internet. It often takes biologists a very, very long time to identify where's the data and really download it. And this simple task can also you can ask the agent to do. And also like CRISPR screen design, cloning protocol design, rare disease diagnosis, which so clinical applications as well, and also like paternalistic data analysis, even on this more computational simulations, like molecular docking, and also single-cell RNA-seq annotation as well, and there are more. And then since then, we inspired by this kind of a wide range of different applications. We really want scientists to feel and touch it and to leverage their own creativities because we can never iterate over the entire task space. So that's why we built a free web platform for scientists at biomni.stanford.edu, where basically in the interface, a biologist would just need to type in what they want to do, and you can upload the files as well. And then the agent would, this is the interface where the main dialogue is happening, and then the agent is basically you can agent will decide if it's a task, it's a simple task like ChatGPT style, just need to directly answer, or it needs to delegate it to like a more time-consuming agent kind of a loop. And then once it's delegated to this, it will perform the reasoning, perform the coding, and also write the visualizations, and then it'll finalize it and return a report to you. And the user can also download the raw Jupyter Notebooks format of this, and then also has many other different features like model selections, and so on. So this is available at biomni.stanford.edu. I think we're basically letting people in every 24 hours. So definitely sign up and then let us know if there's any feedbacks. Cool. So due to lack of time, I will not go into a live demo, but there's also like YouTube videos on how to some live demo on how to use biomni.stanford.edu. Cool. So now what's next? So we're thinking BioOmni kind of unifies the action space, but in real world, there's a lot of tasks that requires more than just actions, and it needs to. If we look at the expert level performance, actually interleaves different actions and reason about it. So we're thinking about if we can train a reasoning agent that can really interleave this action and utilize these actions really well, and then it can potentially generate a completely new way of doing things, right? Because given some verifiable reward, human scientists doing it because it's maybe the first person who just on their mind just come up with this approach, and then just everyone's followed it. But maybe there's a better way to perform the same tasks. So we're very, very interested to see if there's a emergence of know-hows if we train this IO agent, and can we generate know-hows? This can really also inform how biologists can perform their day-to-day research. And we're also really excited about this web lab approach, web lab component, right? Because if we talk about truly AI biologists that can autonomously doing autonomous research, we need a wet lab. We need to do wet lab experiments. So we need to interface the wet lab experiment also with the agent. So connecting the physical and the in silico part of things. So this requires maybe a new programming language of experiments, maybe require robotics and so on. But we think this is a promising approach that we're also kind of exploring as well. Cool. So thank you. This is my talk. I want to thank my authors again. And if you have any questions, feel free to ask me at this email address as well.
Thanks, Kexin, and very interesting talk. And so we have actually a few questions. I have some questions, but let's just start from the chat first. So as the name of your work suggests, Biomni, it basically consider very general task, right? Basically solving the whole biology. So then one question is from the chat, why do you consider such a wide scope of works, and why not focusing on a specific domain? Or do you think that this type of interconnected nature of biology makes it kind of impossible to have, let's say, a very specific, let's say, disease-specific agent or something like that?
Yeah, that's it. Yeah, I think that's a great point. So at the end of the day, we want to get every task to expert level. And then we also want to think a lot about how the biology really use agent. Like I think one way is like there's like gazillions of options of different agents for different tasks, but that's just really hard for kind of biologists to use. Because then the first question is, how do we choose one? How do we choose that? But for general purpose agent, different tasks, you can just like one single chat inbox, and then you can just put one question, and then it will automatically utilize this really well. So that's one idea. And then the second thing is because I think biomedical task is like there's different complexities of task. And even for the same type of task, there's different subtypes of tasks. It's just like it's impossible to iterate over everything. So you can define a specialist agent for each, depending on different granularities you want to go to. So that's why we find this general purpose agent is a much more flexible way. Like it can understand the task, no matter how granular it is, it can automatically figure out the way to do it. And so indeed, like currently, I would say this is a general purpose dynamic agent. So I think at some task, it can actually have some expert level performance, but I think also there's a long tail of tasks that's not on expert level performance. And then how do we kind of incorporate expert level knowledge to these general purpose agent, which I think is the very interesting research direction that we should investigate, and we are also trying to do it right now. Yeah.
Thank you. So the next question is about the verifiability of the agent's proposed actions. So if they have a high rate of hallucinations, do you think that basically the humans to verify the plausibility of those proposed actions or hypotheses takes more time than humans just trying the hypothesis themselves?
Yeah, I think that's a very, very interesting point. So I think it's basically like a like a trust issue, and also basically actually when we released the web platform at first, indeed there's some steps that has some hallucinations, and then human scientists immediately noticed that, and then they say, oh, you have hallucinations, and then after like a few days we just we implement a hallucination guardrail system where at each step it is LLM as a judge where it has common hallucination patterns, and then say if you have detected patterns, you should like rewrite this code. You should not do it. And after that, like no one complains. Like it's just a really, really effective hallucination guardrail systems. And I think this is like a trust thing. Like of course people may still argue maybe there's still some hallucinations, but I think this is like naturally after maybe when we try 5 times, you examine all the traits and there's no issue. And then you kind of gradually trust it. Similarly for cursor, right? At first I had like really doubts on, this cursor writes so much code. And now I kind of stopped checking what is in the code. yeah, I think it's also like a human issue. Of course, we need to make sure the agent is like really rigorous on no hallucination 99.9% of the time. But I think in the end, human will get used to it. So it will be kind of a, it will not have cost more time to verify the agent.
Yes, and one question from my side. Actually, the work was really interesting to me, and that's more like a high-level question. So do you think that, or let's say in the future, if you can basically sort out this hallucination problem, do you think, or what do you think would be the optimal position for human in this system? Do you think that we still need human, maybe something more than running the experiments, let's say when the robotics is more advanced? What would you consider as the right position for humans, which is hard for these agents to replace?
Yeah, that's a great point. Yeah, I think if you see here, we still need it's actually still very sensitive to what the input is to the agent. So I think human in the future we just need to create a lot of this input, the input text to the agent. It's almost like a PI, right? You can just I want you to do this, I want you to do that. Give me it back in a day, and then you can do that now for maybe like 10,000 students, right? 10,000 agents at the same time. Yeah, so I think the human role is basically more like a like a supervisor kind of thing, and of course you will need to verify as well, like the trace and the outcome. Yeah, I think that'll be the I guess the most immediate role of the human. But I also think the agent is gonna, so I didn't mention this, like the agentic systems for hypothesis validation and hypothesis generations. So in the end, it's even possible that the agentic system is gonna automatically also ask the question on how to get this task done. So moving one higher level in the future, maybe humans just need to wait, right? I have this disease. Give me a drug, and then just I don't know. Enter. I mean, this is like super theoretical. It's like it's probably not going to happen in the next few years. But it's like giving some very high level hypothesis. It just goes through every step. It can actually interact with the Biomni agent. It can create some tasks for Biomni agent, and given the Biomni agent output, it can maybe create another task. So yeah, I think definitely we can like continue to layer up the abstraction and the complexity of the task. But also we still need humans, right? Because we need to do validation. We need to really it's at the end of the day, it's the human who makes stuff, right? So yeah.
Okay, great. Thanks a lot again for sharing your interesting work with us. It was great to have you, Kexin.
Cool. Thank you. See you.
Thank you. Okay, and with this, we reach the end of this workshop. We will have some concluding remarks from Patrick. And it was great to be moderating this workshop. And thanks again, everyone, for staying with us. Thank you, and bye from my side.
Thank you so much, Arash, for moderating the session. And yeah, I wanted to just make a few closing statements in addition to maybe sharing with you some of the results that we got out of the survey that we ran prior to the event. And yeah, I just want to share some of the outcomes from that. But this has been a really exciting day, and I think we had a really nice opportunity here to get a cut across the entire field of machine learning and drug discovery, and really seeing all of the exciting directions that are being worked at by the community. So a really, really exciting day, I believe. So I want to dive into some of the questions and the survey results that we've already analyzed for you. And one of the first questions that we've asked in the survey was, what is the main current bottleneck to 10x the impact of machine learning in drug discovery? And we've given you 4 different potential answers. Same type of data in blue, larger scale, same amount of data, but more targeted, higher quality in red, and same data, better models in yellow, and 10x more clinical studies here in green. And we have here, I think, a pretty clear winner. and that people believe that the amount of data may not be the main bottleneck that we're facing, but rather we would like to have higher quality or more targeted data. I think there's a nice second place here, runner-up, where people believe same type of data, but just a larger scale is really what is needed to get us to this level of impact that would be meaningful. With the next question, we've asked you, what's the most underappreciated source of bias in machine learning for drug discovery? And here we've given you the opportunity to answer preclinical models, in vitro, in vivo mismatch, cell line selection, and genetic background, assay conditions, and batch effects, and model architecture and training objective. And I think here we also have a pretty split opinion, actually, between the preclinical models, which are seen as a dominant source of bias in machine learning for drug discovery, but also assay conditions and batch effects, which are seen as a strong contributor to bias. Interestingly, model architecture is ranking the least important in this community survey. The next question that we've asked you is, what kind of model do you believe will have the largest impact on drug discovery within the next 5 to 10 years? And we've given you a lot of different options out of the most popular research directions today in the field. And I'll maybe just go through them in the ranked manner. So the winner in terms of your belief that these models will have the highest impact is actually multi-omics models. I would not have, surprisingly, would have not expected that, but almost half of you believe the multi-omics models will be the biggest contributors to drug discovery from the AI field. I believe the split second ranked here would be protein structure prediction, with around 14% of you saying that would be the largest impact, and another 14% also saying chemical foundation models, for example, the Malformer being another one. And then fourth ranked already the protein language models that we're all very familiar with. Interestingly, here in the lighter shade of blue, general purpose language models do not seem to rank as highly for most of these survey contributors, which interestingly an outcome here that I would not have expected. Which type of organization is best positioned to make the biggest impact in drug discovery and medicine in the next decade? And here we've given you the opportunity. Again, I'll go through them in a ranked manner. Most of you, around 27% of you, believe AI-first startups characterized by being ML native and fast-moving to be potentially the best positioned institutions in this environment. Second ranked, we have biotechs, small and mid-size. Third ranked, according to your survey contributions, would be already nonprofits, with around 17%. And then equally ranked, we have bigger organizations and bigger technology, as well as bigger pharmaceutical organizations. And in the last place, we have the government, actually, which is not ranked and has not been selected by anyone, but second to last being academia here. Interesting results from the survey participants here. Lastly, we've asked you some qualitative questions, for example, which biomedicine papers were you most impressed in the field recently? A lot of you mentioned BOLTS2, virtual cell, remote on-off switching of protein activity, review of large language models and autonomous agents in chemistry, and some contributions from Marinka Zitnik, who spoke today as well, have been highly appreciated. And then we also asked you the converse option. What are some of the papers that you thought were overhyped? And some people mentioned an LLM reasoning paper by Apple and agentic AI paper as well that was brought up by some survey participants. Coming up, some further controversial questions. For example, what will be solved first, AGI or biology? And the predominant fraction of you actually thinks AGI would be solved first. So I assume you guys think AGI will solve biology for us. And then only around 27% of you said biology would be solved, quote unquote, first. Another question we asked you is just what your timeline would be to solving biology with AI. And we've dubbed this bio-AGI and given you the opportunity here to say basically near term, imminent, blue, red, medium term, yellow, maybe perhaps longer term, and then 30 plus years in green, which corresponds to basically probably not in our careers. at least not in mine. And most of you actually believe are split again somewhat. Most of you actually believe it's more of a long-term proposition to solve biology with AI. Around 44% of you said so. And then medium-term belief is around a third of you actually saying that that might happen already within 5 to 10 years. And then, yeah, there's a good group of people that believe it would not happen at all, which is I guess, on the extreme prediction within our projectable timeframes. And then, yeah, there's actually a few optimists that believe it's imminent, with around 15% of you responding that would happen within smaller than 3 years. So very cool answers here, and a nice reflection of the opinions in the field. Next question, what is the most overhyped application of AI in drug discovery today? And again, a very controversial one. We see that for this, most of you actually think all of it is justified, with around 31% voting the lighter blue shade here. Secondly, ranking is AI-driven target discovery, which a lot of you believe is overhyped. Digital twins, I guess the third option that most of you preferred, being overhyped. Foundation models and for biology and de novo molecule generation, I think they share around 10% here, with the remainder of you thinking that these directions might be a little bit overhyped in the field. Coming up next, what is the most neglected but critical component in machine learning-driven drug discovery pipelines? And again, in order of your selections, bridging discovery and clinical translation actually is something that 28% of you thought would be the most neglected part, and could potentially be harboring some unsolved potential here. The second point being mechanistic interpretability, so understanding more about the mechanisms, about how our models make their decisions, being around 24% of you saying that that is a neglected part of the pipelines that we're not spending enough time on. And then robust data curation, negative data curation, 80%. And then with 14% already the fifth ranked human in the loop model design, and last ranked around long-term toxicity prediction and integration with clinical EHR type data. Last question that we asked you is, should machine learning researchers be more involved in? And again, in the order of you guys' beliefs, experimental design, with around 44% of you believing machine learning field should be much more involved in that. Second ranked, real world deployment. I'm happy to see that in particular, around 27% of you believe this is a very important topic that we're not involved in enough yet. And I would say the remainder of the four are around 10%, all equally ranked, with ethical implications, clinical trial modeling, and regulatory conversations, where around 10% each of you believe that would be a topic that more people should be focusing on in the field. So a lot of really cool answers, and very cool to see pulse check of the field here with this survey, and definitely something that I hope gives us a little bit of thought for the future. So it has been a really, really exciting session today, and I really appreciate all of you tuning in today and spending the day with us and our speakers. I think we've had an amazing session, and really a festival, I would say, of AI and machine learning in drug discovery. And yeah, I hope we can see you again in our next instance coming up in the following year. And yeah, thanks so much for attending the session. And the recording, as always, if you haven't been able to make it to all of the speakers' sessions, will be made available, of course, through the YouTube channel once more. So thank you so much for attending, and see you soon at our next instance.